Can you see my screen?
Yes. Yeah, okay. Good. Okay. And then I will test. Yeah. So you can hear me, right?
Yes.
Okay, let me share my screen. Oh my God, I need to do something.
There is a green.
No, I think we can ask the next person to test first because I need to do something on my laptop. Okay. Let me check. Is it okay now?
Yes.
Yeah. Okay. Okay, we stop this.
You can show your full screen.
This one. Is this good now?
Yes.
Okay. Okay, I will create this. How to create this? Stop share. Okay. So next one we also. Let me check who's next one we can. Gerona. Is Gerona here? No, not yet. So this Gerona didn't show yet, right? And Liu Fei is here? No. Yang Jie here? Also no. Chaturvedi here? Oh, only we three now, right? Okay. So let's wait for other participants. Okay, let's wait.
Would you like me to share my screen too?
Yeah. Oh, yeah. Oh, sorry. Just okay. Yes. Yeah, okay. Yeah, and you can start to share.
Is that working now?
Yeah. Working very well.
Good.
Oh, Gerona. Your name is Gerona. But the name showing there is different, right?
Are you talking to me?
Yes.
I am third presentations, Thanapoom Osathanun at 10:45.
Oh, okay. Oh, presentation. Okay. Oh, I was thinking. Okay. Got it. So you are the third one, right? Third one in our session. Okay.
Exactly.
Now for Liu Fei still didn't show. No. Liu Fei, Yang Jie, and Chaturvedi. Oh my God.
Ms. Han, can I just ask, are you the chair for this session?
Yes.
Right. Oh, good. Thanks.
Yeah. Only
Perfect.
with three, Kim and oh, okay. I think another one joined.
And Prof. Jin should be joining as well.
Oh, okay, okay, yeah. Hmm. The other three they didn't show yet.
So it's just, I've been asked to mark 2 of the talks today, but obviously there's more than 2 talks. So I'm just wondering who's marking the other ones or grading. I'm not sure what's going on.
Chaturvedi. Is Liu Fei and Yang Jie here? And Chaturvedi. Oh.
I would just be prepared that maybe they'll join a bit later. They maybe not got the message that you have to join at 10 o'clock.
Okay.
So maybe whoever's managing the room will have to look out for these people during the middle of some of the talks, maybe.
Okay.
Oh, because I never realized there's so many streams in parallel. So there'll always be people jumping backwards and forwards, especially with the symposia going on at the moment.
The other three speakers still didn't show.
I would presume that they will turn up.
Okay.
Hopefully, both from unless the connection to Sichuan is broken.
I hope they won't have any problem about their screen
Oh, here we
sharing.
go. Maybe someone here.
Yeah, yeah. Mhm.
Hi, Prof. Jen.
Good morning, Professor Jane and everyone.
Morning,
Oh,
Risa. Hello.
hi, everyone.
Hello, Lisa.
Prof. Jin, if I can ask you a question, are you only marking 2 talks? Is
Yes,
that right?
only 2.
Right.
Okay, let's start. It's already the start time for Lily.
So should I start?
Yes, yes,
Yeah.
yes. Yeah.
Hello, good morning, everyone. I'm Lily, and my topic is Semoflurane 4D enhances vascular stabilization by recruiting shed. This study is funded by General Research Fund, and there is no potential conflict of interest relating to this study. Let's start from the background. Angiogenesis is a multi-step process, including initial and maturation stage, and the vessel formed in initial stage is unstable and tend to regress. So the recruiting of parasites is critical for vascular stabilization. SEMA4D is an angiogenic factor which can function through plexin B1. It has been reported that SEMA4D could regulate maturation of tumor vessels by recruiting parasites. Shed has angiogenic factor and also can function like a parasite. And the Western results show that shed can express high level of parasite markers, while the mechanism have not been fully investigated. To better mimic the in vivo vascular formation, we adopt this 3D microfluid model, and the layer 3 channels can be seeded with cells. And our hypothesis is SEMA4D could induce the paracrine effect of HUVEC through plexin B1 receptor, and this endothelial-derived factor could further recruiting shed as parasite to support the vessels. Our aim is to examine the effect of SEMA4D in vascular stabilization. First, to elucidate the effect of SEMA4D on the migration, proliferation, and the differentiation of shed. And then to investigate whether SEMA4D could increase the stability of vessels. Here, HUVEC was treated with SEMA4D, and the condition medium was collected and shed seeded in the UPS insert. And the transfer results show that SEMA4D treated HUVEC condition medium could significantly enhance the migration of the shed. Similar results can be seen in the model. Shed seeded in the lower channel and the HUVEC in the upper channel, followed by added SEMA4D. And SEMA4D could significantly enhance the migration speed of the shed. For the proliferation, there is no difference between the condition medium with or without SEMA4D. SEMA4D treated HUVEC condition medium could enhance the expression of Ang2 at 24 hours, and also enhance the expression of alpha SMA and SM22 alpha at 72 hours. And here we use this setting to investigate vascular stabilization. HUVEC was seeded in the central channel and up channel, followed by shed seeded in the lower channel, and SEMA4D added in the up channel. For culture for 7 days and staining. And from the results, we can see that SEMA4D could enhance the total vessel length and also enhance the shed-covered vessels. For higher magnification photos, here we can see that sheds support the vessels. And this we can see a clear lumen, and the sheds support the vessels. To investigate the mechanism, we use SIN to knock down plexin B1 in HuVax and collect the condition medium. And plexin B1 knockdown in HuVac could significantly decrease the migration of shed. Similar results can be seen in the model. When plexin B1 was knocked down in HuVax, the total vessel length and the shed covered vessels was decreased. To further investigate which growth factor responds to this function, we detected several growth factors and found that PDGFBB was increased in SEM40 treated HuVac, and there is no PDGFBB expression in shed. And then we use the PDGFR beta inhibitor in transfer to investigate whether it's through PDGFR receptor. And the results is similar to the previous. And SEM40 treated HuVac could increase the migration of shed, and this effect could be blocked by PDGFR inhibitor. And similar results can be seen in the model. Here, when at PDGFR inhibitor, the vessel length and shed covered vessels was decreased. Here come to a conclusion. SEM40 could enhance the vascular stabilization through plexin B1 receptor and by recruiting shed through endothelial derived PDGFBB. Thank you for listening.
Thank you so much, Lily. And anybody have the questions?
I'll just ask one question first. Yeah, so this is well done, very, very impressive results. Do you think how to further refine the model? There's any limitations you may address in the future?
For this model, because here we use we didn't include the fluid. So in the future, maybe we can add the fluid to show the flow of the vessels, flow of the blood. So here we can also regulate the speed of the fluid and the type of the fluid.
Okay. For the potential clinical application, do you think the anti-inflammation is one of the important concern or not in the future?
Yes, the vascularization is the key challenge in the application of tissue engineering. Yes. And now there are many engineered constructs, but the vessels in this construct are major in the initial stage and is unstable and easy to regress. So this study focuses on the vascular stabilization, and hope in the future we can fabricate successful engineered construct with stable vascular system that can transport nutrition and oxygen.
So in this regard, the model is not only limited to use for the pulp regeneration, yes, also for other tissue regeneration as well, yes?
Yes.
Yeah, that's my understanding. Okay, thank you.
Thank you.
I have a question. Did you like quantify the blood vessels and check if these blood vessels are indeed functional?
Yes, we have quantified the vessel numbers, vessel lengths, and also the shed supported the tubes as parasites.
Okay. Do you check like the patency or the number of vascular junctions that will indicate function of the blood vessels?
Yes, we are testing the permeability of this vessel structure.
Okay, good. Thank you.
Thank you.
Hi, Lily. I have one question. You collected the condition median from the HUVEC, which is treated by the SEMA4D, right? So do you
Yes.
think the condition median will also have some SEMA4D and it can direct work on the shed?
We have the control group. Here, the sample, can you see my screen?
Yes, yes.
Here, we have the SEMA4D treated HUVEC group and the HUVEC condition median group and the only SEMA4D group. The only SEMA4D group, there is no effect on the migration of the shed. And for the Western, we also have the same control group. Yeah, and we also have checked the expression of plexin B1 in shed and the migration of shed in plexin B1 knockdown or without knockdown. And there is no difference, so we didn't put the results on.
Oh, okay, okay. Thank you for your clear answer.
Thank you.
Any other questions?
Maybe ask one quick one. So why did you choose this model design? Were there any other options? And are you thinking of developing this in future studies?
Here for this model, because I have tried the vessel formation on the 2D surface of material, but this is only for maybe for 2 days, and for the longer time the vessel will be regress. So we chose this model can culture for longer time and to study the vessel stability.
But are you thinking of you or your research team? Are you thinking of developing it further?
Yeah. For the future study, we can include more factors to investigate how to make a more mature and more stable vascular structure, and also do some animal study to test the stability of these vessels.
Right. Thank you.
Thank you.
Okay, any other question? Yeah, I just have another question about your animal study that you just mentioned just now. So how will you treat the cells with SEMA4D? You will add the SEMA4D also in the animal model?
Yes. For the simple animal study, we will add SEMA4D together with the cells in the material plug and to see whether it can enhance the vascular stabilization, can produce more vessels.
So you will treat the SEMA4D both on HUVEC and shed?
On HUVEC only. Maybe on shed can as a control group.
Oh, okay, okay, nice, nice. Okay, let's wait for any other question. Okay, if no, you can just stop your screen sharing, then I can start share my screen test.
Okay, thank you.
Thank you for your good, very nice presentation.
Thank you.
Oh, let's chat the, oh, here, okay. Okay, just received one message from Dr. Prasanna. If anybody want to ask a question, they also can put to the question box on the web portal. Okay. So let me start my presentation.
Sorry, if I may clarify, I don't have Lily Zhang on my scoring sheet.
I think we may have different people. I think-
Sorry.
Yes, I have the two here.
Yeah.
Lily Zhang, and then the next one is Hang.
Yeah.
Yuanyuan.
I didn't want to
I
say the
only marked
names.
two.
Yeah.
Sorry. You're
So
saying
I only marked two presentations. Lily Zhang,
I have
and then next one.
5 on my list. There's Han Yuanyuan, there's Liu Fei, there's Kon Su
Oh, okay.
Tisupon. Is it a different? Am I in the right session?
I think so. Yes, I only marked two. Lily Zhang and then Han Yuanyuan.
Okay, I don't know if they send me the
Be
right
okay.
form.
I'm presuming they have. We've all got different selections, but I'm sure
Okay.
someone is there masterminding everything.
Ah, I see. Okay,
Yeah.
I
I'm
got
not
it.
sure, but
Okay.
Rolly, how we mark? Just as 1, 2, 3, 4, 5, yes? Up to 10.
And no, it's in the scoring sheet. Have you been sent the scoring sheet?
I have the scholarship 40%, 30%. How to mark? Put the percentage or put the mark 1 to 10.
Put the percentage in because it adds up. It's an Excel, so it generates the mark at the end.
Well, what do you
Type
mean?
in-
Then the first one, for instance, I mean, 70%, 80%, 40%, like this.
No, type in like a percentage of 40. So type in 25, 30, 35, 40.
So 40 means so I put in, for instance, 36, like that.
Oh,
Yeah,
to do this,
I
huh?
used to use. We obviously some of the people we're scoring are listening, but yeah, I think I always put in numbers of 5, but there's no reason you can put any number you want in there. Up to 40, then up to 30, then up to 30, and it will.
Oh, okay.
add
You put
together.
for instance,
I think you
36,
put the
like
number.
this. Yeah?
Yes, put
Yeah.
in,
Oh, okay.
yes, 12, whatever.
put
something.
a number 1 to 10. Okay.
Not percentage. Okay, just a number.
A number
Okay.
out of 40, then a number out of
Oh,
30,
okay,
then a
okay.
number out of 30. It will add up to 100.
Oh, okay.
So it'll give you a percentage of 100 once you've typed in everything into the boxes. I think they used the marking sheet at a previous thing.
Okay. So
Sorry, Miss Han.
yeah, it's okay. So everything clear now? Should I start?
Yeah.
Yeah,
I
okay.
think so.
Hi, good morning. I'm Yuanyuan Han, a PhD student from the University of Hong Kong. The title of my today's presentation is HIF-1 stabilization enhances odontal osteogenic properties of shed in vitro and in vivo. As we all know, stem cell-based therapy is a promising strategy in tissue regeneration. There are various dental stem cells which are available for stem cell-based tissue regeneration. Among all the dental stem cells from human aspirated deciduous teeth, which we call shed, is considered as an ideal cell source for dental pulp regeneration due to its easy acquisition, ready availability, self-renew with high proliferation, and multilinear differentiation potential. It has been shown that shed could regenerate dental pulp-like tissue with an odontal blood layer, blood vessels, and nerves in vivo. However, the main challenge for dental pulp regeneration is a harsh hypoxic microenvironment with insufficient oxygen and nutrients at the transplantation site. Especially the dental pulp canal only have one small apical orifice. Hypoxia in dental pulp canal influences the cell survival and following tissue regeneration after stem cell transplantation. Hypoxia-inducible factor 1 alpha, HIF-alpha, is a key transcription factor in hypoxia. When the oxygen concentration is normal, or we usually say normoxia, HIF-alpha will be rapidly degraded by PD2 and VHL-mediated ubiquitin proteins. However, when oxygen is low, which we always call hypoxia, PD2 activity will be inhibited since oxygen will act as a cofactor for PD2. and this PD2 initiates degradation of HIF alpha. Therefore, HIF alpha is stabilized under low oxygen conditions, and it interacts with HIF1 beta and activates the expression of downstream target genes, including vascular endothelial growth factor, VEGF, which assists in promoting and increasing oxygen delivery to a hypoxic region. In our previous study, we have demonstrated that HIF alpha dependent VEGF secretion plays an essential role in including the angiogenic response by promoting the proliferation and the recruitment of host vascular endothelial cells and differentiation of dental stem cells into endothelial cells, both in vivo and in vitro. This paper is published on Journal of Dental Research, and one of our picture was selected as a cover image of that issue. In addition to vascularization, odontogenic differentiation and dentin-like tissue formation are also essential in dental pulp regeneration. Some studies have proved that HIF alpha also play an important role in osteogenesis, so that we hypothesize that HIF alpha stabilization also could help dentin-like tissue formation in dental pulp regeneration. Therefore, the aim of this study is to examine dentin formation of HIF alpha stabilized shed in dental pulp regeneration. And our objectives are first to stabilize HIF alpha expression in shed through PD2 knockdown. And second, to explore the effect of HIF alpha stabilization on odontal osteogenesis of shed in vitro. And third, to demonstrate the odontal osteogenic effect of HIF alpha stabilization of shed on dental pulp regeneration in vivo. First, we knock down the PD2 by lentivirus to stabilize HIF alpha in normoxia. The knockdown efficiency was checked by Western blot. As we can see in the figure, PD2 expression was significantly decreased compared with the control group, whereas HIF alpha expression was significantly increased. Then we induced control shed and PD2 knockdown shed using osteogenic induction medium to evaluate the effect of HIF alpha stabilization on osteodontogenic differential shed. Staining for early odontal osteogenic marker ALP after 7 days of induction confirmed a significantly higher ALP activity in the PD2 knockdown group compared with control group. Subsequently, alizarin red staining revealed that a significantly higher levels of mineralization were detected in the PD2 knockdown group after 21 days of odontal osteogenic induction compared with control group. Later, we performed RT-PCR to evaluate the MRA expression of some odontal osteogenic markers. As we can see in the figure, the DSPP and DMP1 were elevated following HIF alpha stabilization, although there was no statistical significance. At the same time, ALP and RUNX2 MRA levels were significantly increased after PD2 knockdown compared with the control group. Western blot results show the expression of DSPP, DMP1, ALP, and RUNX2 proteins was significantly upregulated in HIF alpha stabilized shed following odontal osteogenic induction. To examine the potential use of HIF alpha stabilized shed on dental pulp regeneration, we used a full-length tooth fragment in vivo mouse model. Single root human teeth was sectioned and prepared with taper reamer. Then the coronal end was sealed with MTA. Later, control shed and PD2 knockdown shed were encapsulated in 0.5% purimetric peptide hydrogel and injected into the root canals. After 24 hours, these tooth fragments were implanted subcutaneously into the dorsum of 6-week-old male skin mice. Tetracycline hydrochloride was injected at day 10, 15, 19, and 24. The samples were retrieved after 28 days. and 2 slides were examined for tetracycline staining. These tetracycline fluorescences revealed a clearly defined fluorescent line here in the dentine of the 2 fragments injected with PD2 knockdown group in contrast to the control group. Quantification of fluorescence intensity confirmed a significant difference between the 2 groups. This indicates more dentine-like tissue formation on the interface between the 2 dentine and pulp-like tissue in the PD2 knockdown group. Then we did IHC for DSPP. This showed the positively stained cell layer here, cell layer lining the dentine pulp interface, indicating an odontoblast-like cells. PD2 knockdown group showed a significantly higher positivity for DSPP in the odontoblast-like cells, which also appear to have processes extending into the dentinal tubules compared to the control group. So our study proved that HIF-1α stabilization could enhance the odonto-osteogenic differentiation of shed in vitro, and also can promote the dentine formation in pulp regeneration model in vivo. However, the signaling pathways involved in HIF-1α stabilization induce the odonto-osteogenesis maybe need further research. So in conclusion, HIF-1α stabilization could significantly enhance the odonto-osteogenic differentiation of shed and promote dentinogenesis during pulp-like tissue regeneration, both in vitro and in vivo. Thanks for your time and welcome for questions.
So you're chairing your own questions. I could maybe start, this is quite far away from my sphere of knowledge. I'll just ask something very sort of fundamental. For HIF-1α, how does it respond to different oxygen tensions? Is there a cutoff? I mean, between how hypoxic does it need to be?
Yeah, I just mentioned here this PD2 will degrade this HIF-1α, and when it works, it needs the oxygen to act as a cofactor. So once oxygen is very low, so this PD2 activity will be inhibited. So that's why the HIF-1α cannot be degraded by PD2. Then HIF-1α will have stabilized expression.
Right, but my question was at what concentration of oxygen? Is it a sharp gradient? Because there's a lot of hypoxic environments in the oral cavity. I just wondered, is it known? Is it consistent in different cells? HIF-1α, is it 10%, 2%? How hypoxic is your hypoxia?
Yeah, this hypoxia, according to the papers and studies, when the oxygen concentration is lower than 2%, we usually call it hypoxia.
Right, thanks.
Can I have one quick question?
Yeah.
I just wondering, usually the hypoxic condition, we use it for culture iPS cells to maintain it in stemness state. But your results suggesting that hypoxic condition induce differentiation into odontoblast. So how could you explain this conflict of the results, something like that?
Okay, I hope I got your point. For this one, at the beginning, why we choose HIF-1α stabilization, just because when we transplant the cells in vivo, the environment quite harsh. So that's why we just hypoxic these cells to make it kind of adapted to the environment in vivo. So that's why we choose this HIF-1α stabilization. And as you said, for the LPS, right? You already use for
the iPS, induced pluripotent stem cells. Normally, we culture iPS cells in hypoxic condition to maintain it in the undifferentiated state, meaning that the hypoxic condition will suppress differentiations, but your results show the opposite way. So you have
Oh,
experience on
okay.
that.
Oh, okay. Yeah, that might depend on different cells. That IPS cell, or maybe you will have different a region, right? And also, I think the HIF-alpha stabilization won't only work on the stem cell that maintenance, stemness maintenance. So in the differentiation, especially the bone regeneration, is very well studied for this HIF-alpha function.
Thank you.
I have one comment for you to consider here. So Lily just presented the interesting data, yes, regarding the SIMA4D the model. Do you think there is a possibility to combine both together to synergize the result and for the future potential clinical application?
Yeah, it's very interesting to combine these two together. We already try something, but according to our previous data, the HIF-alpha stabilization won't work with that SIMA4D, or maybe the not a very significant link with these two factors.
Okay, so you, one more question is just for curious. So how about the HIF-1 beta? So there's any role to play as well?
The HIF-1 beta won't change according to the oxygen concentration. It always be there in the nucleus. So once HIF-alpha is stabilized, then they can work with, they can interact with the HIF-1 beta.
Okay, thank you.
Thank you.
Can I ask what would be your next step if you want to translate this to the clinic?
Next step, actually this is a very basic research. Later, if we want to apply this in the clinic, we need to find a good way to stabilize HIF-alpha, because now we are using this lentiviral to stabilize HIF-alpha. It's not applicable in the clinic. So later we need to find a better way, maybe combine with scaffold or some small molecules to stabilize HIF-alpha.
Thank you.
Thank you. Okay, any other question? If no, I will quit this screen sharing and welcome for next speaker, okay?
Thank you. I'll go.
Shall I share presentation now? Is it my turn?
Yeah, it's yours.
Okay, thank you. Let me share my screen. Sorry about that. All right, hope everyone can see my screen properly.
Yeah.
Today I will present to you the research topic entitled Mechanical Force Regulate Induced Pluripotent Stem Cells Behaviors. In general, oral structures, for example, parental ligament, subject to the force all the time. The force from chewing, the force from orthodontic treatment, the force from traumatic occlusions. And this force regulate various types of tissue change and modulate tissue homeostasis. Different type of force, for example, compressive force, tensile force, or shear force, or direction of the force, even the magnitude of the force, can differently modulate cell functions or cell behaviors. Our lab focused a lot about mechanical force and the response of cells, especially stem cells. We have studied both in terms of the force control stemness and also the force control differentiation, particularly on orthogenic differentiations. For the stemness, we study in stem cell isolated from human exfoliate deciduous teeth, and we apply the force to these cells is induced expressions of IL-6 in both protein and mRNA level. And this IL-6 will further activate JAK pathway and releasing of ATP, and this ATP bind to P2Y receptors and cause the upregulation of REX1, which is a pluripotent stem cells marker, meaning that when we apply force to the chest, it can maintaining this cell in stemness conditions. However, we also study the role of force to induce orthogenic differentiation in osteoblasts and also PDL cell as well. We apply the force to osteoblasts, and this force will release the ATP, and ATP will further bind to the P2X1 receptors and cause the upregulation of Wnt3a in both protein and mRNA level as well. Wnt3a will further induce beta-catenin accumulation and translocate into nucleus and cause the upregulation of OSX, which is the Octerex, the transcriptional factors of the osteoblast differentiations. On the contrary, when we study in the PDL cells, we apply the same force. It also upregulate the ATP release as well, but this ATP did not have any functions on orthogenic differentiation in the PDL cells, but rather this force can upregulate the intermediate molecule, which we not know yet, and this intermediate molecule will further upregulate TGF-beta expressions by two receptors and cause the upregulation of oxalic and subsequently promote orthogenic differentiation. So you can see that we apply the same force type, same magnitude to this different cell type, but it's induced although the end, the same phenomenon, but the mechanism is different. That leading us to study the role of force in the 3D culture of iPS cells by using the global gene expression analysis using RNA sequencing technique. For first, we try to culture iPS cells in 3D construct and drive it into mesodermal lineage. We isolate iPS cells and culture it in floating conditions as an embryonic body. And after that, we add retinoic acid to induce these cells to commit into mesodermal lineage. And after that, we adhere that cell to the culture plate. Further, we subject these cells to the mechanical force for 24 hours under serum-free condition medium. As you can see that you can see 3D cell construct in both control and force condition. When we apply the force to these cells, it will not alter any cell viabilities. So after we subject cell to the force for 24 hours, we isolate the total RNA and sequencing those RNA and try to identify which gene is upregulated or downregulated. From the high throughput RNA sequencing, we found that about 900 genes are upregulated and about 1,000 genes are downregulated. And we categorize those together and do the enrichment analysis using bioinformatic analysis against KEGG pathway, and we found upregulated genes mostly involved in cell cycle regulations by induced various of pathway. However, the downregulated gene mostly related to the metabolism pathway. So to further analysis the role of mechanical force on cell cycle, we identify those genes related to cell cycle, and we found that most of the genes upregulate when we apply the force to the iPS cells. And this was also confirmed by using real-time polymerase chain reaction. So we found that CCNG1, CCNG1, and CDK6 is upregulated compared to the control. So when further, we subsequently identify the functions of how this upregulated gene is involved in cell cycle progression. So we do the same setting. We culture the cell in floating conditions, adding retinoic acid to induce the mesodermal commitment, adhering for 24 hours, and culture in serum-free condition medium, and subject to the force for 24 hours. And after that, we remove the force out and culture in normal growth medium for 48 hours to allow cell to progress in cell cycle. As you can see here that from the cell cycle analysis using flow cytometries, we found the downregulations or the lower or decrease of G0, G1 subpopulations in those treated with force. And also we found the increased numbers of S phase and G2M phase in those cells treated with force as well. So overall, when we calculate the proliferation index, we found the upregulation of proliferation index in force treated iPS cells. Further, we investigate how the force control apoptosis, because in the 3D construct, as is commonly induced cell apoptosis, which is a very burden for those iPS cells that we culture in vitro. So we identified 350 pathway, which is a pathway that involved in apoptosis, and we further do the apoptosis assay using annexin B and potassium iodine staining. And you can see here that when we apply the force to the iPS cells in 3D construct, the force can reduce the number of cells that have the early apoptotic markers, which is have lower of the annexin B positive populations, but it will not have any influence on late apoptotic cells or necrotic cells. So from this result, we imply that the force that we apply to the iPS cells in 3D construct can induce cell proliferation, but attenuate cell apoptosis, especially the early apoptosis event. Then we further try to identify which pathway may be involved in this mechanism of how force controls cell proliferations. One of the pathway that we thinking of is the TGF-beta, because from our previous study, we show a lot of cell when we apply to the force, it can activate TGF-beta secretions. So this is what we thinking of is might be happening in the same way as iPS cells that we study in this present experiment. So we further confirm the expression of TGF-beta by subject cell to the force, and we found that the TGF-beta genes upregulate at 24 hours, and also the protein upregulation was confirmed by ELISA technique and also ICC staining technique as well. To prove that TGF-beta might be involved in this mechanism, we just simply use the preliminary study by adding inhibitors of TGF-beta receptor SB431, 542, which is the TGF-beta ALK5 inhibitors. We added inhibitor 30 minutes before we apply the force to the cells. And after that, we culture for 24 hours to collect the proteins and genes to do the PCR analysis and the Western blot analysis. As you can see here that when we add the inhibitors, it can attenuate the effect of force-induced CCND1 and CDK6 expression. So it's likely that TGF-beta may be involved in the mechanism how force can induce cell proliferation in iPS cells 3D construct. So what we are showing here today is that I show you that the force that we apply to the iPS cell in 3D construct can upregulate TGF-beta expressions, and that TGF-beta expression further control the expression of CCND1 and CDK6, which is the key protein or key molecule in cell cycle propagations. And maybe we think that this regulation is further control cell proliferation in this setting. So lastly, I will thanks all funding agency and people in my lab, and also the collaborations that we have with this experiment with Tohoku University. So thank you very much. I'm ready for your questions, if you have any.
Thank you, Dr. Thanaporn, for a great presentation. Can you, from your findings, can you tell us what is the critical implication?
It's maybe not related to clinic yet, but the burden for those people studying iPS cell is that when we culture in 3D construct for further applications, these cells cannot grow anymore, or even apoptosis, because it's the nature of culture in 3D construct. So when we found that the force can modulate these two functions of the cells in culture, so I think it's very interesting in terms of approach or technique that we can use in future culture conditions to promote proliferations and reduce apoptosis in this 3D construct of the iPS cells. And in this 3D construct can be further applied as the cell source for regenerative treatment in future, for example, the bone regenerations.
can I ask maybe a very basic question? So some of my colleagues have been doing work with oral cells with changing the forces applied to them from orthodontics. Can you tell me a little bit about, is it a compressive force? And are different types of forces used for different sets of cells? And do they have different results? Like your mouse IPS cells, you have to be very specific with the forces or the pulses of forces you apply. Do you get different results when those are changed?
I think it's very difficult question to answer. I mean, because like I mentioned, or you said in the questions that different cell type, even we apply the force, but the same force, like compressive force, but we apply differently, like static compress or intermittent compress, is induce different response. So we need to fine tune the specific conditions for a specific cell type in order to control a particular cell response. For example, if we apply the continuous compressive force to PDL cells, it can upregulate rank ligand, which induce the bone resorption, induce the osteoclast differentiation, and further leading to bone resorptions. But when we apply the intermittent compressive force, it also induces rank expression, but as well, but the amount of induction level is lower compared to the continuous compressive force. So meaning that the chewing force, when we do the intermittent chewing, it has compressive force to the PBL. It not have that much influence on the bone resorption, but when we do the static compressive force, like on the orthodontic force, it have more bone resorption. That might be explained related to clinic. So I don't know whether I can answer your question, but that's the best I can respond to your question.
So it's certainly suitable for my level of understanding. Thank you.
Okay, if no other question, let's welcome for next speaker, Liu Fei. Liu Fei. Okay, please share your screen. Your microphone is muted. Yeah, it's okay now. Could you please share your screen?
Hello.
Yes, please share your screen.
It's my turn.
Yeah, it's your turn now.
Okay.
Can you please share your screen? Okay. Okay, please start.
You can see the screen.
Yes.
Okay.这个怎么放上去了？看不见。
你说演讲怎么？
不是，我看他好像没看到屏幕的。
就是共享屏幕。
啊，
然后共享。
不一样。
Could you please start your?
Okay. Could you see my screen?
Yeah, I can see your screen.
Oh,
okay.
It's
not on the show. Can you please just start at slideshow?
就播放。
Sorry, please wait a minute.
Okay.
Oh.
Yeah, it's okay now.
Okay.
Yeah. Please start.
Okay. Good morning, everyone. I'm Filiu, a research assistant from Western China School of Stomatology at Sichuan University. First of all, I'd like to express my sincere gratitude to the organizing committee for providing me such a for this opportunity to share my study here. What I'm going to talk about today is the expression profile and the mechanism of the non-coding RNA and mRNA in the trigeminal ganglion associated with trigeminal neuropathic pain. My report mainly includes the 3 parts. This study was supported by the following findings. Trigeminal neuropathic pain belongs to the first classification of orificial pain, according to the first edition of International Classification of Orificial Pain, published last year. It is attributed to lesion or disease of trigeminal nerve. For example, some orificial clinical therapies, such as wisdom teeth extraction and implant surgery, sometimes damage trigeminal nerve injury and resulting orificial sensory impairment. Some patients may suffer from dysphagia, hyperalgesia, and anhedonia. However, there is still a lack of persistently effective treatment for trigeminal neuropathic pain. The changes of molecules, receptors, and activation or inactivation of intracellular spinal pathways in the trigeminal ganglion play a vital role in the trigeminal neuropathic pain. Also, such knowledge has been obtained, and many potential targets have been found in the trigeminal ganglion. The mechanism of trigeminal neuropathic pain are also poorly understood. Non-coding RNA are not translated into proteins, and many include long non-coding RNA, circular RNA, and microRNA. Now, more and more evidence suggests that they are associated with a variety of diseases through interaction with RNA, DNA, and proteins. Emerging evidence suggests that non-coding RNA are associated with neuropathic pain. For example, non-coding RNA in the dorsal root ganglion and spinal cord were significantly changed under the condition of neuropathic pain induced by spinal nerve or sciatic nerve injury. However, it is known about the role of non-coding RNA in the trigeminal ganglion associated with trigeminal neuropathic pain. Therefore, in the present study, the classical trigeminal neuropathic pain model induced by canonic constriction injury of infraorbital nerve were first used to determine the differentially expressed non-coding RNA and mRNA in the trigeminal ganglion using whole transcriptome sequencing and bioinformatics analysis. First, the mice were randomly divided into sham and CSI group. The CSI mice showed anhedonia from day 3 to day 28 after surgery. On day 7 days after the surgery, the ipsilateral trigeminal ganglion were epicyclated to determine the differentially expressed non-coding RNA and mRNA by RNA sequencing. The results indicated that 216 non-coding RNA, 14 circular RNA, 67 microRNA, and 595 mRNA were differentially expressed. In addition, the 421 differentially expressed gene encoding proteins were determined. In order to validate the reliability of the RNA sequencing results, one of the differentially expressed non-coding RNA and mRNA were randomly chosen to be verified by RT-qPCR. The results were consistent with the RNA sequencing. In order to predict the relationship of differentially expressed non-coding RNA and genes with trigeminal neuropathic pain, a list of 804 non-pain related genes were referenced. Targeted prediction analysis indicated that 107 differentially expressed long non-coding RNA were predicted to target 35 differentially expressed mRNA associated with pain by co-expression. In addition, 3 differentially expressed mRNA or predicted to be regulated by the same differential expressed linker RNA by co-expression and co-location. In addition, two differentially expressed mRNA are predicted to be regulated by two differentially expressed circular RNA. Furthermore, 30 differentially expressed microRNA were predicted to negatively target 8 differentially expressed mRNA, including 7 pin-related differentially expressed microRNA and 4 pin-related differentially expressed mRNA. Geoanalysis of differentially expressed non-coding RNA indicated that most of the enriched geo terms of differentially expressed non-coding RNA and differentially expressed microRNA targeted genes were similar. The significantly enriched molecular function of differentially expressed microRNA were binding related. KG analysis of the differentially expressed non-coding RNA indicated that MAPK signaling pathway, RAP1 signaling pathway, and retrograde and cannabinoid signaling pathway were significantly enriched, which were related to reception. KG analysis of differential expressed genes also showed that several reception-related signaling pathways were significantly enriched. In order to further integration the function of the differential expressed genes with trigeminal neuroplastic pain, a list of reported 1,000 protein-protein interactions involved in pain were referenced. A PPI analysis indicated that 30 upregulated differential expressed genes were involved in pain-specific PPI network, and the Hoover genes were related to nerve injury, regeneration, and proprioception signal transmission, and anti-nociceptive signal transmission. According to the list of the pain-related genes, 39 differential expressed genes were non-pain genes. And the string network analysis of the 39 differential expressed pain-related genes indicates that it showed an interconnected network, including several functional clusters, which were involved in tumor necrosis factor signaling pathway and neuropeptide signaling pathway, long-term depression, glutamate-gated calcium ion channel activity, and peptide hormone binding. In addition, nerve injury-related genes such as ATP3 and MP9 were also included in the network. It is well known that non-coding RNA and circular RNA can regulate the mRNA through the competing endogenous RNA regulation. Therefore, the cRNA network of the differentially expressed non-coding RNA were also constructed. In the cRNA network of non-coding RNA, the targeted differentially expressed mRNA were related to pain, such as CTSD and LMX1B. Similarly, in the cRNA regulatory network, several pain-related genes also involved in the targeted differentially expressed mRNA. In the primate study, the differentially expressed non-coding RNA and the circular RNA, microRNA, and mRNA in the trigeminal ganglion of CCI mice were firstly reported to be associated with trigeminal neuroplastic pain. And several receptive signaling pathways were significantly enriched for differentially expressed non-coding RNA and genes. And the differentially expressed non-coding RNA in the trigeminal ganglion may play a vital role in the development of trigeminal neuroplastic pain through cRNA regulation. The results of this study provide a basis for the studies about the role of differentially expressed non-coding RNA in the trigeminal ganglion neuroplastic pain, and bring a new insight into the novel potential neuroplastic target and the mechanism of trigeminal neuroplastic pain. Finally, I would like to express my sincere gratitude to all the members in the team of Professor Division, and thanks for your support from West China School of Stomatology. Thanks for your watching. Hello. Hi.
Hi, is there any question? Yeah, I would like to ask you one question. So
Okay,
you
thank
did very good job about this analysis of non-coding RNA. So what is your plan for the further research? Which one you will pick up to do some experiment?
Yes, thank you for your question. In the parental study, we just show the knowing or noted the differentially expressed genes, and most of the non-coding RNA are noted. So in the future, maybe we will focus our attention on the novel differentially expressed non-coding RNA or genes. Also, in the parental study, we just take into account the differential expressed genes associated with pain genes, but maybe we can find some new genes related to the pain in the future. So now our team screen the target by PCR and Western blot to find the stable expressed or stable upper bracket expression of long non-coding RNA, circular RNA, and microRNA. And now we have found several interesting targets. So our further study will do experiment to prove this target non-coding RNA in the trigeminal neuropathic pain.
Okay. Thank you for your answer.
Thank you.
Okay, let's move on to next speaker. Yang Jie. Is Yang Jie there?
Hello. Can
Yeah,
you hear me?
so Liu Fei, could you stop sharing your screen?
Okay.
And then so that Yang Jie can start her. Okay.
Could you please wait for a minute?
Okay.
Okay. Okay. Hello everyone. My name is Jie Yang, a graduate student from West China School of Stomatology, Sichuan University, and this is my mentor, Professor Jie Fisher. I'm glad to be here for this presentation. My topic today is chemical CXCL1 and its receptor CXCR2 in trigeminal ganglion contribute to nerve injury-induced orofacial mechanical angina. The content of this presentation is divided into 3 parts: the background, the methods and results, and the conclusion. Why I choose this item? I think it can be illustrated from the following 3 parts. First, the trigeminal nerve injury is a common and serious complication of various dental procedures, such as molar extraction, dental implant surgeries, and maxillofacial surgeries. And this may result in orofacial neuropathic pain. In many cases, the pain can spread to adjacent areas that are innervated by the uninjured nerve branches, which is called orofacial ectopic pain. Nociceptive stimuli to the orofacial region are typically conveyed to the trigeminal ganglion, and the TG is mainly composed of TGNS and SDCS. Our previous studies have demonstrated that a series of molars and receptors participated in the interaction between TGNS and SDCS, which is the key potential mechanism underlying neuropathic pain. Second, chemical CXCL1 and its receptor CXCR2 participated in neuropathic pain while ganglion neural interaction in the spinal cord. Third, the research of Takara has suggested that VGPCs participated in mechanical angina induced after trigeminal nerve injury. Moreover, the PKC signaling pathway is involved in the expression and function of VGPCs. However, with the CXCL1 and CXCR2 in TG, involved in the mechanic adenina after genital nerve injury, and whether expression of VGPCS through PKC is largely unclear. Next, let me introduce our methods and results. In general, our research can be divided into two parts. The first part aims to investigate whether CXCL1 and CXCR2 in TG participated in the regulation of mechanic adenina after INX. We transected the inferior abdominal nerve to establish pain model, and this figure showed the process of INX surgery. Then we tested the mechanic hypersensitivity in the vesical area innervated by the infraorbital nerve to study mechanic adenina. In addition, the drugs were directly injected into TG under the guidance of stereotaxic apparatus, and this figure showed the injection and validation process. After INX or sham surgery, the HWT was tested on day 1, 4, 7, 14, and 21, and the results showed that compared with the sham group, the INX treated group displayed distinctively lower HWT at day 1 until day 21 and reached its lowest level on day 4. This indicated that INX triggered mechanic adenina. However, when we injected elective and of CXCR2 or as shown in this figure, compared with the sham group, to further explore whether CXCL1 was sufficient to trigger mechanic adenina in naive mice. Recombinant CXCL1 was directly injected into TG, and the HWT was tested 30 minutes later. And the results demonstrated that the HWT of CXCL1 treated mice were significantly lower than that of the sham group. However, when we pretreated or PKC inhibitor injection, the CX PKC inhibitor. According to part 1, we conclude that INX and exogenous CXCL1 can trigger mechanic adenina, and it was altered by inhibition of CXCR2 and PKC signaling pathway in TG. As for part 2, we focused on the expression. We focused on the distribution and expression of CXCL1, CXCR2, and its downstream moleculars in TG after INX. First, we investigate the distribution and expression of CXCL1 in TG, and the results demonstrated that CXCL1 was distributed in both TGNS and SGCS, and compared with the sham group, the INX mice exhibit significantly greater mRNA and protein expression of CXCL1. As for CXCR2, the results showed that CXCR2 was expressed in TGNS, but not SGCS. And the MR and protein expression of CXCR2 were upregulated too after INX. In addition, we further analyzed the protein expression of PKC and PPKC. Compared with the sham group, the INX significantly increased the protein expression of PPKC from day 1 to day 7. However, after inhibiting CXCR2, the INX-induced upregulation of PPKC was reversed. This may indicate that CXCR2 participated in the activation of PKC after INX. Furthermore, we analyzed the MR expression of the 7 major VGPCs after INX. We found the mRNA levels of these VGPCs in INX mice were lower than that in the sham group. However, only the expression changes of Kv1.4 and Kv1.1 were consistent with those of CXCR2, as the all changes from day 1 to day 14 after INX. So we further analyzed the protein levels of Kv1.4 and Kv1.1. The WB results confirmed that the protein levels of Kv1.4 and Kv1.1 were downregulated after INX. However, when we inhibited CXCR2 or PKC in TG, the downregulated mRNA and protein expression of Kv1.4 and Kv1.1 were upregulated. This observation indicated that CXCR2 regulated the expression of Kv1.4 and Kv1.1 through the PKC signaling pathway after INX. According to part 2, we can conclude that CXCR2 inhibited the expression of VHPCS through the PKC signaling pathway after INX. In concluding, this study revealed that the activation of CXCR2 in TGNS triggered mechanic endothelium after trigeminal nerve injury by downregulating the expression of Kv1.4 and Kv1.1 through the PKC signaling pathway. The findings shed new light on the mechanisms underlying the development of orofacial ectopic pain and suggest that CXCR2 might be a potential therapeutic target. In the end, I want to appreciate my mentor and my whole team members for their kindly support. And thank you all for your patience. Thank you, that's all. Hello.
Yeah, hello. Thanks, Yang Jie, and for your nice presentation. Is there any other question from others? If no, let me ask one small question. So this CXCR2 is one receptor for this CXCL1, right?
Yes.
Yeah, and this CXCR2
it's the main receptor of CXCR2.
Yeah, okay. Yeah, so this CXCR2 is also one receptor for the CXCL2. So why you choose this CXCL1 or CXCL2? Is there any different function between these two things?
I have because CXCL1 and CXCR2 was studied in the central nervous system, and they found CXCL1 and CXCR2 was participated in mechanic and was participated in the neuropathic pain after nerve injury, and the CXCL1 was expressed in ganglion, and the CXCR2 was expressed in neuron, and they participated in pain through the ganglion-neural interaction in the spinal cord. But the function of CXCL1 and CXCR2 in TG was not unclear. So I choose this item.
Okay.
That's all.
Okay. Thanks for your answer. Is there any other question? Okay, so please let me ask one more question. So what kind of clinical application will your research do? I mean, how could you apply this in the clinic? I mean, is there any clinical significance or application about your research?
Yes. In clinical, we found the trigeminal nerve injury is a common complication in various dental procedures. And these procedures may induce orofacial ectopic pain. And according to this study, we found CXCR2 might be a potential therapeutic target for this clinical pathologies. So I think it is helpful for our clinical treatment.
Okay, thanks. Very nice research project and very nice presentation. Let's move to our last speaker, ChatVD. Is ChatVD there? Yeah, ChatVD Susie Sapon.
Yes.
Yeah.
So I share slide now, right?
Yeah.
Okay.
Okay, so could you please share your PowerPoint?
That's just a moment, please. Sorry. Do you see my slide, right?
Yes. I can.
Yes. Good morning, everyone. My name is Chatwadi Kongsuthi Sapon. I'm a PhD student in Oral Biology program, Chulalongkorn University. So today, I'm very pleased to share my project, which is the Ticlusive Enhanced Odontogenic Differentiation in Human Dental Pulp Stem Cells. Okay, so let me start with the background of the study. So in case of the dental cavitation, normally pulp capping is used to initiate the dentine formation. However, major concerns for current material are degradation failure, and sometimes it can cause inflammatory reaction, which leads to several unsatisfactory outcomes. When we look a little bit deeper in terms of the cellular response, it's required a cascade of the stem cell activation, proliferation, migration, and differentiation to the new odontoblast-like cells that will come into the reparative dentine formation. So our research focus was to enhance the osteogenic differentiation capacity of dental pulp stem cells, which will promote the dentine reparative formation. And this pulp injuries study show the importance of Wnt in the dentine regeneration. So by using the Wnt transcription factor transgenic mites, we can see the increase of the Wnt expressing cells and also co-expression with the Wnt expressing cell and the PCNA positive cells, which is the proliferative marker, suggesting that the Wnt beta-catenin signaling is activated in response to the pulp injury. And also we can see the co-expression of the AXIN2, which is the Wnt target genes, with the DSPP, indicating that the odontoblast-like cells are the Wnt active. So to promote the dentine repair in the tooth presented with the dental pulp, dental cavitation, Wnt beta-catenin might be a promising target that can be modulated to enhance dentine regeneration. And then the next question would be how we can modulate the Wnt signaling pathway. Sorry, I cannot. Normally, Wnt has the lipid group which make the Wnt protein hydrophobic and decrease the stability of the Wnt. But with the Wnt, with a small molecule GSK3 beta antagonist, the beta-catenin destruction complex will lose their ability, allowing the beta-catenin to accumulate and translocate into the nucleus for further Wnt signal transduction. And the Ticlusive is a GSK3 beta antagonist that is commercially available and has been developed into the medication for the Alzheimer's disease treatment. So we aim to employ this substance to develop as its of use target as a pulp treatment. So the beneficial effect of Ticlosip has been previously described. However, the intracellular mechanism on dental pulp stem cells remain unresolved. So the aim of our study was to investigate the influence of the Ticlosip on the human dental pulp cell response, and further the molecular mechanism of Ticlosip-induced differentiation will be elucidated by the RNA sequencing technique. So first we collect the primary pulp cells by tissue explantation and subsequently treat with a range of the Ticlosip concentration that correspond with those previously described. And this is the brief workflow of our study, and we expect to apply this knowledge for development of the Ticlosip-based bioactive material for the pulp capping. So next is the results and discussion of our study. So first, the isolated pulp cells were characterized as stem cells by showing a positive mesenchymal stem cell related surface marker and the absence of the hematopoietic stem cell marker. And also they show the multi-lineage differentiation towards the osteogenic differentiation and also the apoptogenic differentiation. And also we show that Wnt pathway was activated by the Ticlosip treatment. So we stain the beta-catenin by immunofluorescence, and we show that all of these concentration can increase the subcellular localization and also the nuclear beta-catenin nuclear translocation compared with the control. And in terms of the cell proliferation, we found that Ticlosip treatment did not significantly affect the cell proliferation, the subpopulation in cell cycle progression, or the colony forming units ability of the dental pulp stem cells. And next, we evaluate whether Ticlosip involved with the apoptosis. So although we found that the significant upregulation of the anti-apoptotic genes was found in the Ticlosip treatment group, but the average fold is less than twofold. And by using the flow cytometry, we found that there was no difference in the early or late apoptosis among the groups. So altogether, we concluded that Ticlosip had no effect on the apoptosis. And in terms of the cell migration, results show that Ticlosip did not increase the percentage of the wound closure at the 48 hours after the treatment. And regarding the osteogenic differentiation, we found that the Ticlosip can enhance the alkali phosphatase staining and also the mineralization, although the 200 nanomolar Ticlosip group exhibit just a little increased mineral deposition. And this result corresponds with the osteogenic related genes expression, which show that in the 200 nanomolar group did not really significantly affect the osteogenic related genes. In contrast, in the 100 nanomolar, it can significantly promote the osteogenic related genes, the RUNX2, the ALP, and also the BMP2. And also in 50 nanomolar groups, it can increase the DSPP mRNA expression. So according to our result, we found that Ticlosip did not affect the cellular response, which we speculate that it might be from the cell type specific. In contrast, the Ticlosip can enhance osteogenic differentiation of the human dental pulp stem cells. And because the Wnt transcription factor, it occupies the RUNX2 promoter. So when we activate Wnt, it leads to the upregulation of the RUNX2 expression, thereby stimulating the osteogenic differentiation. And because 200 nanomolar didn't significantly upregulate the AXIN2, as you can see from this graph, so we suspect that this concentration might not really effectively to activate the Wnt signal, leading to less potency than the other concentration. And this table is the top 25 differentially expressed gene in the Ticlosip treated human dental pulp stem cells. And some genes are involved with the osteogenesis. For example, silencing of this fibroblast growth factor 5 can decrease the osteogenic differentiation of several mesenchymal-related stem cells. And also silencing of this APC downregulated 1 genes. It can decrease the mRNA expression of the beta-catenin and also the osteogenic differentiation of dental follicle cells. And this table shows the top 10 KEGG enriched pathways in the taglicib-treated human dental pulp stem cells. So our results correspond with previous studies which show that GSK3 can modulate several metabolisms. And also several previous studies show the interaction between this signaling pathway with the Wnt, for example, the interleukin 17 and also the tumor necrotic factor alpha. So in conclusion, our in vitro study can demonstrate that the taglicib could activate the Wnt beta-catenin signaling, thereby promoting the osteogenic differentiation capacity. So in case of the pulp cavitation, this concept could be developed by incorporating the taglicib into the capping material to stimulate the osteogenic differentiation capacity of dental pulp stem cells, aiming to repair the dentine pulp complex naturally. So, but there is still a need for further study, such as delivery system that can incorporate the taglicib and delivery these small molecules to a specific area that need to be repaired. And also the characterization of release and biological activity, which is very crucial for developing this taglicib-based nanoparticle for clinical practice. And special thanks to IIDR Incorporation and my faculty and also Dental Stem Cell Biology Research Unit. Thank you.
Hi, thank you for your presentation. Very interesting project. So I just have one small question. So this taglicib can work, I think can work very helpful in the clinic dental capping procedure, right? So is that this one commercial or easily available in the market?
Yeah, it's actually is a medication that already commercially available in the market, but it's not really for the pulp capping yet. It's still for the, because when it's related to the Alzheimer's disease and several neurological disorder. So it's this medication is in the like phase 2 clinical trial for the Alzheimer's disease, but not yet for the pulp capping.
Oh, okay. So will you do some more further research, combine this one with dental capping materials?
Yes, maybe I plan to for the further study, maybe because the taglicib is still we cannot really control the release because it's hydrophilic, it has hydrophilic activity. It is like just other growth factors. So maybe we need to incorporate to the nanoparticle so we can we can specify the area that we want to want it to action, like in the capping.
Okay, okay. I think it will help a lot if it can work very well in the clinic.
Hopefully.
Thanks. Is there any other question from the audience? Okay. Okay, let me ask you one more question. So this taglicib will work on the Wnt signaling. So will any other signaling that you analyzed will also be involved or be researched later in your project?
Well, because it's actually there's several signaling pathways that related to the dentine regeneration. So actually in our lab, in project, and our colleague, they also interested in notch signaling, which is actually very interesting because it's like it's more cell-to-cell contact, so it might be quite appropriate for being developed to the pulp capping because we want we want to like scope the area that we want to make the dentine regeneration, like in just that area that has the like odontoblast, cell death or something like that. There are quite a lot of cell signalings apart from the wind.
Okay. Okay. Except the dentine formation or osteogenesis, will this also can work on some angiogenesis or nerve formation, neurogenesis? Is that
Yeah.
that?
Yeah, I think because in the previous literature, they also describe about the effect of the wind in enhancing the angiogenesis and also in the neurogenesis as well. So that might be another aspect, another interesting aspect that we can evaluate. Yeah, apart from just dentine regeneration.
Yeah, very nice.
Yeah.
Yeah.
Thank you for your
Thanks
advice.
for your, thank you for your presentation. Yeah.
Okay.
So any other question? If no, then our session is almost done. So thanks for all the audience and all the speakers. I hope all the speakers in our session part already done the work, right? Yeah, I hope no one lost. Yeah, so okay, so that's all. That's our presentation. Any person come to help me to finish this Zoom meeting? Risha, are you there?
You calling me?
Yeah. So our session is done now.
Yes, good. Thank you for all participation.
Okay, so we can leave this meeting now, right?
Yes.
Okay. So thanks for everybody for your attendance and for your presentations. See you.
Bye-bye.
Bye-bye.
So good afternoon, everyone. I will be the chair of this session this afternoon. So before we start, I would like to make sure that everyone can share the screen and your audio and video work appropriately. So we have 6 presenters this afternoon. Maybe we start from the first one. So Ms. Gupta, can you try to share your screen and then speak a few words so that we know that we can hear clearly.
Okay, sure.
Okay. So you try to share
my screen now.
your
One second.
Do you have your PowerPoint slide?
Yes, sorry, one second. Yes, can you see my slides?
It's loading. It's good. Okay, perfect. So maybe let's try the second one. Dr. Ali. Dr. Ali, are you here? Easton? Dr. Easton? Are you with us? I think you are muted. Can you unmute yourself?
Okay, can you hear me right now?
Yeah, sure. So can you share,
Okay,
try to share your screen?
Yes. Can you see my screen right now?
Yeah, perfect.
Okay.
Then you can stop sharing now. And let's try the third one. Dr. Carnell. Is Dr. Carnell here? Or maybe we try the first one first. Dr. Wairiwan.
Yeah, hello.
Hello. Yeah, we can hear you.
Okay, I'm gonna start to share my screen.
Like turned up your voice a little bit.
Is it visible? Yeah, can you see my screen?
Yes.
Okay, thank you.
Good. Okay, now let's try the next one. Dr. Hendriwan.
Yeah, I'm here. Can you, is my voice audible?
Yes, clearly.
And I'll try to share my screen now. Is it visible as well?
Yes, very good. Thank you. And then the last one, Dr. Devan. He's not here?
Dr. Devan is coming.
Okay. So our session would be starting at 3:15. Okay.
Okay, so Dr. Devin is here.
Dr. Steven, can you hear us?
Okay, sorry.
It's okay. So please try to share your screen, see if it works.
Okay. Wait a bit. Can you see my screen?
Yes, very good.
Okay. Can you see it changing?
Yes.
I think that's enough.
Okay.
Thank you.
Then we have one more, Dr. Kano coming.
So hello, Dr. Yu. I'm Yuan Chen. I'm the judge of this section.
Hi, nice to meet you.
Yeah. So I just want to know, so they have already know they have to present 10 minutes and keep 5 minutes for question. All the presenter they already know, right?
I think so.
Thank you. Okay, so will you make a timer to remind them, or you will
Yeah,
set
I
a
will.
timer? Okay, thank you very much. Okay, so I just listen and ask a question. Okay, thank you very much.
Very good.
Very nice. Thank you.
So Dr. Kano just left the room, right? So I think we can start now. Good afternoon, colleagues and students. Welcome to the oral session, the oral microbiology and the immunology session. So today we will have 6 presentations in these sessions. So each presenter will have 15 minutes to present, including 10 minutes PowerPoint presentation and 5 minutes Q&A session. So hope we can all have a fruitful discussion this afternoon. Now let's welcome our first presenter, Ms. Gupta from the University of Hong Kong. So Ms. Gupta, please share your screen.
Thank you so much. Okay. Should I start now?
Yes, please.
Okay. Did you know that microbial biofilms cause up to 75% of human infections? And in the context of oral health, dental biofilms are implicated in dental caries, mucosal infections, root canal infections, and periodontitis. Microbes within a biofilm show greater resistance to antimicrobial agents and greater virulence as compared to their planktonic counterparts. So what are biofilms and why are they so hard to eradicate? Microbial biofilms are highly functional, complex organizations of microbes which are encased and protected by extracellular polymer matrix. It may be the water within the biofilm and the EPS, which protects and prevents drug penetration into the biofilm, thus making them so hard to eradicate. Moreover, there could potentially be genotypic and phenotypic changes which contribute to their resistance. So currently, the primary focus remains on finding safer molecules that prevent biofilm formation in the first place. We rarely talk about oral biofilms without mentioning enterococci. E. faecalis and E. faecium are 2 of the most common enterococci species causing human infections. E. faecalis is a virulent organism predominantly found in chronic root canal infections. It is known to be a pathogen that resists several treatments and is a model organism to study root canal infections. E. faecium, on the other hand, is a virulent pathogen which causes multiple infections. For example, urinary tract infections, endocarditis, bacteremia, and meningitis. E. faecium is among the so-called escape pathogens which are notorious for antibiotic resistance. These pathogens are highly virulent and are known to cause one of the deadliest hospital-acquired infections with mortality rates as high as 40 to 42%. Their multi-drug resistance is among the top 3 threats to global public health. In the last few decades, there has been a surge in antibiotic resistance caused by the widespread use of antibiotics, which kill planktonic cells. The ability to form biofilms by bacteria is considered to be one of the most efficient escape mechanisms from antibiotics. Thus, currently, a widely accepted therapeutic approach is to use sublethal concentrations of drugs that inhibit biofilm formation rather than killing planktonic cells. Enterococci are intrinsically resistant to a number of first-line antimicrobial agents. At present, there is no definitive approach to tackle enterococcal infections, and this poses a global challenge. Good afternoon, everyone. My name is Gunjan Gupta, and today I'll be sharing our work on biologically derived surfactants on Enterococcus biofilms. So biosurfactants are non-toxic, biodegradable, amphipathic molecules. These contain a sporous hydrophilic head and a fatty acid hydrophobic tail. Their unique chemistry allows them to reduce the surface tension between two immiscible liquids of varying polarities. These biomolecules also exhibit a wide spectrum of desirable properties, such as emulsification, detergency, solubilization, anti-cancer, antiviral, and antibacterial properties. These could potentially revolutionize the field of biomedical science. They have also been used to improve the bioavailability of hydrophobic drugs. One of the most commonly studied biosurfactant is sophorolipid. Sophorolipids are extracted from non-pathogenic yeasts. Sophorolipids are glycolipid surfactants consisting of two components, lactonic and acidic. The lactonic component is responsible for the antimicrobial action. However, it is extremely hydrophobic and it requires the acidic component to increase its bioavailability. The acidic component acts as a detergent. It has better solubility and foam production. Crude sophorolipid is a mixture of lactonic and acidic forms. The ratio of these two forms depends on the culture conditions and fermentation times. For this study, we used crude sophorolipid with lactonic to acidic ratio of 75:25. We obtained it from our collaborators from the National Chem Lab located in Pune, India. So despite the substantial body of research, biofilm modulation and eradication without damaging host tissue remains a critical concern with contemporary antimicrobial agents. Moreover, the effect of biosurfactants has never been tested on enterococci before. So we hypothesize that sophorolipids exhibit significant antibacterial activity and thwarts biofilms of enterococci. The aim of this study is to investigate the effects of sophorolipids on E. faecalis and E. faecium planktonic cells and biofilms. To assess the effects of crude sophorolipids on planktonic killing, we performed the standard broth microdilution method. The lowest drug concentration, which displayed inhibition of bacterial growth, was established as the MIC value. To assess the biofilm inhibition by sublethal drug concentrations, we performed the standard crystal vial assay. The absorbance was measured at 570 nanometer to determine the MBIC. So we derived three primary results from this experiment. First, 250 microgram per ml of crude sophorolipid was established to be the MIC value for both E. faecalis and E. faecium, as it inhibited planktonic growth. Second, 250 microgram per ml of crude sophorolipid inhibits 90% biofilms for both E. faecium and E. faecalis, as we can see from the graph below. Third, sub-MIC concentrations inhibited 25 to 50% biomass in E. faecium, but was not able to prevent biofilm development in E. faecalis. The ever-increasing resistance by pathogenic microbes to current antimicrobial agents has shifted attention towards newer, safer therapeutic agents such as biosurfactants. Biosurfactants are able to disrupt membranes, leading to cell lysis and metabolite leakage. They can disrupt protein conformations of important membrane functions such as transport and energy generation. and they form a film that changes the wettability and surface energy of original surface, affecting the adhesion properties of microorganisms. In the present study, we explored antibacterial and antibiofilm effects of sophorolipids. From this study, we can conclude that sophorolipids kill planktonic cells and moderately inhibit biofilms of Enterococci. Since these are FDA-approved molecules, further research can lead to rapid commercial development of biofilm inhibitors. This brings us to the end of this presentation. I would like to extend my heartfelt gratitude to the Faculty of Dentistry, HKU, and the lab technicians Becky Chung and Joyce Yao for their support and guidance. The funding for this project was granted under the Undergraduate Research Program Scheme, the Faculty of Dentistry, HKU. Thank you so much for having me here today. I will be taking questions now.
Thank you, Ms. Gupta, for your nice presentation. Now, may I invite questions from the audience to Ms. Gupta? You can type it in the chat box or you can send me directly, both okay. Or you can just present your questions. So may I have one question for you?
Yes.
This is a pilot study, right? So do you have any comparison with other common antimicrobials?
We didn't really, this is a pilot study, so it was just a proof of concept whether these actually show any antibacterial and antibiofilm properties. So I didn't actually look into to compare it with other antimicrobials right now. But yes, I can look into that.
Okay. So how about the toxicity of these sophorolipids?
So far from the previous researches that I've read about sophorolipids, they're supposed to be non-toxic and they're biodegradable and they're completely produced by non-pathogenic yeasts. And for other kinds of sophorolipids are also produced by microorganisms. But I think we need more research into it to see the toxicity as well. Yes.
Okay, thank you for your answer.
One question. All right. How would this, if it's materialized, help to improve management of diseases related to biofilm?
If it is materialized, like this is still a very initial part of the research, I think it could potentially prove to be very useful in all of medical field. For example, there have been some researches surrounded around how if you can, because ephesium causes a lot of nosocomial infections in hospitals. So there has been a lot of research around if these biosurfactants can be coated on catheters and instruments to prevent biofilm formation on those instruments before subjecting them to patients to prevent infections. And in the field of dentistry, I think if we could look into how these biosurfactants can be used to maybe coat root surfaces for periodontitis patients, maybe in the form of mouthwashes, maybe it could be used to enhance the effects of other antimicrobials like chlorhexidine. But yes, we definitely need more research for that. And we could also use it in the field of endodontics. Maybe we could use it while performing root canal treatments as well.
Thank you.
Thank you. So we have one more question from Dr. Ali. So he asked about your future direction of the research.
Okay, so right now we have like a lot of some limitations to the study. For example, we did not analyze the exact mechanism of how biosurfactants work in inhibition of biofilms. So we would like to look into that in the future. And also we only tested the ratio of lactonic to acidic from 75 to 25. We can also potentially change the ratios to see which one exhibits the maximum antibacterial and antibiofilm effect. Also, we only tested like enterococci, and oral infections are usually multiple species. So it would be useful to look at other species as well. Thank you.
Thank you. So do we have any more questions? If not, let's thank Mr. Gupta for her nice presentation. And now let's move on to our next presentation. So Dr. Ali, please share your screen.
Okay. Sure. Okay, can you see my screen now? Can you see my screen?
Yes.
Okay, nice. Okay, good morning, everyone. Sorry, I'm just because right now in my home country, just 9 a.m. So, and good afternoon for my colleagues everywhere in Southeast Asia region and in Hong Kong. My presentation today is entitled as Strain-Dependent Virulence Profile in Enterococcus faecalis Clinical Isolates. So I will continue with the story that Gupta has started with the Enterococcus faecium, but I will talk about the other member of Enterococci, which is Enterococcus faecalis. And as Gupta mentioned that Enterococci, these are a very common commensal pathogen that exist in the gastrointestinal tract of the human and animals, but it also implicated in the pathogenesis of various chronic and life-threatening infections like infective endocarditis and surgical wounds, urinary tract infections, and bacteremia, and also persistent infections that affected the dental root canal systems. And one of the most prevalent species of the Enterococci, which is implicated in all these kinds of infection, is the Enterococcus faecalis. And the reason for the involvement of this pathogen in a wide range of infection is related to its virulence determinants. And let me tell you that the virulence is the ability of the microorganism to cause damage to the host tissues or to cause a disease. And Enterococcus faecalis has a lot of factors that enable it to destroy the host tissues and cause the diseases, and to evade also the host immune response. For example, Enterococcus faecalis has a wide range of the proteins that located on the cell surface that enable its adhesion to the host cells. It also secretes the 6 hormones, which are implicated in the exchange of the antibiotic resistance genes and the surface adhesion and the surface adhesin genes. Also the lipoteichoic acids, which cause the exaggeration of the inflammatory response of the host tissues. And it secretes some lytic toxins such as cytolysin and the proteolytic enzymes such as gelatinase. And both of these will be the focus in my presentation. But let me tell you also more about that virulence profile of Enterococcus faecalis. This was a very hot topic in the past few years and in the past decades to be explored and investigated. That's because, as I said, that these virulence profile of Enterococcus faecalis explain its implication and its involvement in wide range of infection. Therefore, numerous research groups have started to explore the virulence profile of different isolates of Enterococcus faecalis and from different environmental sources and from clinical sources as well. Out of the virulence factor, which I have previously mentioned, I focused on 2 of them, which are the cytolysin and the extracellular gelatinase or proteolytic enzyme. And let me tell you that the production of these 2 virulence factors are regulated by a chemical communication process, which is called quorum sensing. So this process is simply, it's a kind of communication or a social interaction between the microbial cells, and it is regulated by chemical molecules which are called autoinducers, and it is related to the cell population density. With respect to the role of these factors in the pathogenesis of Enterococcus faecalis mediated infection, let me tell you that the cytolysin is, as a toxin, is an antibiotic toxin which causes membrane lysis and degradation of the cell membrane of the host cells, such as erythrocytes and polymorphonuclear leukocytes and macrophages. Also, it destroys the retinal epithelial cells and the intestinal cells. Therefore, the research has proved that the presence of the cytolysin and the secretion of this toxin is associated with a higher morbidity of the enterococcal infection. The other factor, which is the extracellular gelatinase, it has also a role in the pathogenesis of E. faecalis mediated infection because it causes degradation of various host tissue proteins like the collagen, dentin, fibrin, and fibrinogen, and complement protein. Therefore, it contributes to the spread of inflammation and infection and the evasion of the host immune response. It also contributes to the formation of biofilms. And as Jupiter said, that the biofilms, these are microbial aggregates which protect the individual microorganisms from the effect of the antimicrobial. And the gelatinase has been shown to contribute to biofilm formation in Enterococcus faecalis by breaking down the cell membrane of the cells and enhancing the release of the DNA into the biofilm matrix. So the production of gelatinase is enriching the biofilm matrix and thus increasing the resistance of Enterococcus biofilm to the effect of antibiotics and the antimicrobial agents. As I said, also, like the degradation of the host tissue proteins and cells by gelatinase has resulted in increasing the level of the tissue destruction and increase the spread and dissemination of infection. So why do we study all these factors? The studying of these factors is central in the development of the novel therapies to tackle the Enterococcus faecalis mediated infection. And this is very important in the current era because we are exploring we are now witnessing the reduction in the effectiveness of the antibiotics. And we are looking for a novel therapies to tackle the infection because there is a reduced production of new antibiotics. And targeting this microbial virulence factor is essential because it is associated with a lower risk of enhancing the microbial resistance. And it also allow us to explore a novel target and the mechanism in bacterial cells to develop a novel therapies. Also, it has a minimal effect on the normal flora of the human gastrointestinal tract in contrast to the conventional antibiotics, which might increase the risk of opportunistic infections. Therefore, the objectives in my study was to investigate the quorum sensing regulated cytolysin and gelatinase profile in the clinical isolates of Enterococcus faecalis. And this is the scheme of this study. So we have a collection of Enterococcus faecalis strains, which has been isolated from failed root canal treatment with persistent lesion in Sweden. And we first confirmed the identity of these strains using a rapid identification method using colony PCR and using the EDA genes primers because the EDA gene was found to be specific for the Enterococcus faecalis among the gram-positive bacterial isolates. After the confirmation of the species identity, we also explored the presence of the genes which encode the production of the cytolysin and gelatinase, and also the genes which control the expression of the FSR quorum sensing. And the FSR quorum sensing, this is the system which regulates the production of gelatinase. And finally, we explored that the two phenotypes regulated by the cytolysin and gelatinase, and these phenotypes are the hemolytic activity and the proteolytic activity. And this was detected on the blood supplemented agar or for the hemolytic activity. and the gelatin supplemented THA for the detection of the proteolysis. So the species identity has confirmed that the strains that we have all belong to the Enterococcus faecalis, as we see in the agar-agar images. When we explored the presence of the cytolysin A, the cytolysin A is the activator of the cytolysin. So this is a gene which encodes the production of protein, which converts the cytolysin into the active toxin, which is directly involved in the tissue destruction and degradation. And by the screening of the isolates, we found that only 18% of the isolates have a positive cytolysin A gene. For the GEL-E gene, we find that around 88% of the isolates contain the GEL-E gene, as we see in the images. For the FSR genes, we have 3 genes, because all these genes belong to the same system, which is the FSR system. So we have the FSRA, FSRB, and FSRC. And surprisingly, we find that the same ratio has existed in all these genes. We find that 42% of the isolates contain the 3 genes, and 58% were negative for the FSR locus or the FSR genes. When we explored the phenotype, if you find, like on the image on the top left, here we can see the hemolytic activity. As the arrow shows here, like there is a clear zone which is surrounding the bacterial colonies. This is indicator of the positive hemolytic activity. This means that the bacterial cells are secreting the hemolysin, which causes a breakdown of the blood which exists in the agar plates. And on the right side, this is the proteolytic activity, which is controlled by gelatinase. We observe it here, like the opaque zone surrounding the individual colonies, and this is indicator of the positive proteolytic activity. And by the screening, we find that the positive hemolysis was only observed in the 18% of the isolates, while the positive proteolytic activity was observed in the 42%. And this was very interesting, that although the strains have like around 88% of gelatinase, only 42% show the proteolytic activity. And this is because some of these strains only contain the GEL-E without containing the regulatory system, which is the FSR system. So that's why we see that the 42% is here is showing the proteolytic activity, and 42 and the same isolates carry the FSR gene and also the GEL-E gene, which means that the phenotype is expressed in the FSR positive and the GEL-E positive strains. Let me tell you about the limitation that exists in this pilot study, that only the phenotype has been tested on the routine agar plates in the lab, and it might be better to explore and to test the extent of the virulence in a more in vivo model to show how these virulence genes or the virulence factor contribute to the pathogenesis of the infection, and also that the limited number and the clinical sources of the Enterococcus faecalis. And in the future, we might consider testing these phenotypes and the genotype from a wide range of the strains and from different clinical sources. So we can finally conclude that the presence of these virulence factor in Enterococcus faecalis isolated from the root canal treatment failures could suggest the potential role of these factors in the pathogenesis of the endodontic infection and persistent post-treatment infections. Thank you, and now I can receive your questions, please.
So thank you, Dr. Ali. Do you have any question for Dr. Ali?
Okay, may I have a question?
Yeah, sure.
Okay, so thank you very much for the nice presentation. So you mentioned you collected 33 E-faecalis strains. So how many strain? I mean, the strain is different or are similar. All these strains is similar in genotype.
Actually, the strains have been collected in Sweden by a group of collaborators in Sweden, and those strains has been isolated from different patients, and those patients has seeking the dental treatment for the root canal retreatment because the lesions, those teeth was associated with persistent infections. But those strains are different from each other in terms of the genotype. What we have did is to see that the genes or the variance profile genes in these different strains. So the strains are different from each other in terms of the genotype and also the expression of the related phenotype.
Okay, I see. So how you decide the simple number? Because in total you collected 33. So now this project is undergoing. You want to collect more samples, clinical strains.
Actually, we in our research group we had a huge collection, like around 60 strains of Enterococcus faecalis, but these strains has been isolated from different geographic locations and from different clinical sources. And the future direction in the research will be to expand the examination of the virulence profile in the entire group, on the entire collection of the strains, and to see the prevalence of these genes in the entire collection and correlate it with the source, because as I said that we have those strains from different countries and from different populations. So yes, we need and we also seeking to collect the strains from other countries and from other clinical sources.
Okay, thank you very much.
Thank you.
I have a question for you here, actually, because you are studying on the clinical isolates, yes?
Yes.
One fundamental issue here is we now know that the host or microenvironmental condition will affect the microbial profile, including these species, I believe. So I think, first of all, you need to provide, if possible, the clinical data, the individual profiles of the patient with the failure to end the treatment. That may give additional information here, because according to your results, the isolates will present with different profiles, yes? In genetic expression, whatever. So this is an important point. That's why not surprising to see the variation among the isolates, yes?
Yes.
Because you detect the gene for this and for that, it's different, yes?
Yes.
So that's one point. Another one is just read the title of the presentation. You just talk about the variance profiles. I thought you cover a lot of different variance factors. Actually, just focus on the colon-scenting related molecules, yes? So, so in this regard, maybe more specific, just point out, okay, in the title. Otherwise, are you going to extend to the study to really investigate the other variance factors of this important species, such as and also the whole cells?
Yes. I think that the future direction in our research group that to explore more and in depth about the virulence genes, or to test the other virulence factors like the surface adhesions, the expression of the the lipotecaleic acid related genes, on all these things in the huge collection of the strains that we have. So yeah, that's a very good point to work on the future.
Okay, thank you.
Yeah, thank you, Prof.
So thank you, Dr. Ali. We overrun a little bit. So now let's welcome our next presenter, Dr. Kano.
Sure.
Yeah, please share your screen. Thank you.
Is my screen visible now?
Yes.
Okay, thank you. Should I start now? Okay, good afternoon, ladies and gentlemen. First of all, I'd like to thank the IADRC team for the opportunity that has given to me to present our research about the effect of Mauli banana on dual species Caries bacteria biofilm. So this was an in vitro study that was conducted in University of Lambung Mangkurat, Banjarmasin, Indonesia. And let me, Diora Susabila Farno, present the research. We have no conflict of interest in this research. Our research comes from the background of the high cases of caries in Indonesia that has reached a number of 88.8%. The caries itself started by the adherence of Streptococcus mutans with its virulence product of glucan. As we know, as Streptococcus mutans is the initiator bacteria in pathomechanism of caries.
Sorry, Dr. Kano, I think we are now still in the title page.
Okay, let me change the screen, sorry. What about now?
Now it's in the background. Oh, yeah.
Okay, thank you for reminding me. Now I'm going to continue, but our background is come from the high cases of caries in Indonesia. That has reached a number of 88.8%. The caries itself started by the adherence of Streptococcus mutant as the initiator with its virulence product of glucan. The glucan, as the metabolite of Streptococcus mutans, will be used by Lactobacillus acidophilus to colonize and form a dual-species biofilm. A dual-species biofilm is a biofilm that consists of two microorganisms and has more virulence than a monospecies biofilm. In this day, the prevention of caries is done by the use of chlorhexidine 0.2%, but it has a lot of negative side effects, and that's why a local herb as an alternative is observed. One of a local herb that has been used as an antibacterial is Maoli banana stem. It's a species of banana that contains a lot of tannin, saponin, alkaloid, flavonoid, isoleucine, and also cinnamic acid. The biocomponents of the Maoli banana will all together work against dual-species biofilm formation. The study aims to analyze the effect of Maoli banana stem gel, or MBSG, on the dual-species biofilm mass of Streptococcus mutans and Lactobacillus acidophilus. The method of this research observed the percentage of the remaining dual-species biofilm mass and the percentage of reduction in dual-species biofilm mass. The research group consisted of 12 groups. Group 1 was given 0.2% chlorhexidine gel as a positive control. Group 2 was not given any treatment, which is as a negative control. Group 3 was the media control, and Group 4 to 12 were given MBSG concentration. All groups were incubated for 72 hours and then stained with crystal violet and observed with a micropipette reader. This is the result from the research. As we can see here, the higher the concentration of the Maoli banana stem gel, the less the remaining of dual-species biofilm mass. The asterisk mark on the screen indicates that it has a lot significant difference with the control positive, which is chlorhexidine 0.2%. This result indicates that Maoli banana has an ability to reduce the biofilm mass formation, but it hasn't passed the ability of chlorhexidine gel as the golden standard to reduce the biofilm mass. This is the second result, which is about the reduction of dual-species biofilm mass. As we can see here, the higher the concentration of Maoli banana stem gel that has given to the group, the higher the reduction of dual-species biofilm mass. But still, as the previous result shows, that this result can pass the chlorhexidine gel of 0.2%. From the discussion before, we have said that MBSG has a lot of biocomponents that will work against dual-species biofilm formation. First component that we will talk about is about isoleucine. Isoleucine will act as one of the antimicrobial peptide that has an electrostatic activity and also will able to block the cellular communication of the bacteria itself. The electrostatic activity of isoleucine will bind to precursor of peptidoglycan in bacterial, and it will cause a disturbance in membrane permeability, and it will cause a degradation of the cytoplasm. The degradation of cytoplasm itself will cause the synthesis of nucleotide acid decrease, and it will lead into the disturbance of biofilm mass formation. The electrostatic activity between isoleucine and bacterial cell will disturb the adherence of the biofilm and will lead into the biofilm mass formation disturbance. Isoleucine also able to block the cellular communication that will decrease the regulation of quorum sensing or the communication system that is used by the bacteria to communicate to each other. And this is the second biocomponents in MBSG that is able to be an antibiofilm, which is cinnamic acid. So cinnamic acid has a number of TPSA of 37.30 angstrom. It indicates that these molecules has a good way to diffuse the penetrant membrane. It will penetrate the membrane by passive diffusion, and it will blockade the hydrogen ATPase, and it will cause acidification of the cytoplasm. Once the cytoplasm got acidification, it will denaturate the protein of the bacteria and will disturb the formation of the biofilm itself. And then in molybdenum stem gel also has alkaloid that will blockade the enzyme of topoisomerase, and it will act as the competitive inhibitor of quorum sensing. It will blockade the initiation of biofilm formation and cause a biofilm formation disturbance. Also, there is tannin that will disturb some of important enzymes, which is synthesis of reverse transcriptase and DNA topoisomerase, and also the glycosyl transferase. Glycosyl transferase itself is used by the Streptococcus mutans to produce glucan, and is used by another bacteria to colonize. And then this will make an intersection of cell envelope transport, protein and adenosine, and it will lead it to the disturbance of biofilm formation. The next thing in the MBSG is saponin. It will cause a membrane cell disturbance and will disturb the protein regulation of the bacteria itself and make the biofilm formation get disturbed. Also, the last thing is flavonoid. The flavonoid will able to block the synthesis of glycosyl transferase. Also will block the DNA gyrase as an enzyme. It will cause an intramembrane leakage and will make the biofilm formation get disturbed. As we can see on the buffer graphic, we can say that the negative control, which is dual species biofilm of S mutants and L acidophilus with no intervention, has a highest mass of remain dual species biofilm. It is because the negative control has a proper environment and nutrition to perform the activity of metabolism. It will provide the good environment for S mutants and L acidophilus to co-aggregate, and then the metabolism will keep increase. And also about the chlorhexidine gel 0.2% is still being the best agent as an anti-biofilm. The ability to reduce biofilm mass of chlorhexidine 0.2% is better than MBSG. Chlorhexidine 0.2% itself will able to disturb the cell wall of the bacteria, and it will make a precipitation of the macromolecule to cytoplasm, and it will make the cell wall of the bacteria get ruptured. And then the chlorhexidine 0.2% itself will able to penetrate into the cell membrane and will make the inactivation of ATPase, and it will make a disturbance in replication. Once a replication in bacteria get disturbed, it will cause the bacteria won't be able to produce a lot of virulent stain, and it won't be able to co-aggregate with another bacteria. So the conclusion about this research is that MBSG or molybdenum stem gel has an effect on the dual species biofilm mass of Streptococcus mutans and Lactobacillus acidophilus in the form of a decrease in dual species biofilm mass. Thank you. I would like to discuss this matter with all of you.
Well, thank you, Dr. Cano. Do you have any question for Dr. Cano?
Okay, so
can I have a question? Sorry, you can go off if you want.
It's okay. No problem.
Okay, I just have one brief question for Dr. Cano. So your results has reported that 0.2% chlorhexidine is showing a better effect on the biomass than the MBSG gel, right?
Yes.
So so what do you think the reason for that? And how can you improve the effect of the MBSG gel to be comparable to the conventional treatments that we have been using currently?
Okay, thank you for the question. So our researchers, we made a gel, actually, we made our own gel based on the extract of molybdenum stem itself. And then we add some substitute like for the gel. And our hypothesis is that the ability of the MBSG is not as great as the chlorhexidine is because the gel itself hasn't reached the good constituent itself. So we have to improve our constitution at the gel substance in itself.
Okay, so the gel consistency, you mean with that is the reason for that? Okay.
Yeah, like the HPMC and then the glycerin itself, like the basic ingredients for the gel.
Okay, is there any reason for your selection of this specific plant to prepare the extract?
Okay, so molybdenum stem, this plant is our area is a famous plant and is already used as an antibacterial. So there is a previous research about this that has stated it could prevent the metabolism of Streptococcus mutans. So this is a continued research.
Okay, thank you.
Okay, so actually my question is similar with Dr. Ali. I just want to know, do you know what's the relationship of Streptococcus mutans and Lactobacillus acidophilus, because you choose these two bacteria, right? Do you know their association? Are they beneficial to each other? For example, they have some synergistic effect for growth, or they will inhibit the growth of each other. Do you know do they have some synergistic or inhibit effect between two bacteria?
Okay, thank you for the question. So as we know that Streptococcus mutant is one of the initiator in pathomechanism of carriers, and Streptococcus mutans is able to produce the glycosyltransferase enzymes. This enzyme has a lot of virulence factor that will be used by Lactobacillus acidophilus. And there was a previous study that stated the same results with this. It was said that the dual species biofilm of S mutans and L acidophilus will work synergistically to perform the carriers itself. So yeah, the relation between S mutant and L acidophilus is less synergetic.
Okay, thank you.
Thank you for the question.
You concluded that the MBSG gel has some impact on reduction of the biofilm mass, yes? So
Yes.
my question here is, because in your study, you set up how many? I think 9 testing groups. That's a little bit unusual, because you tested from very low percentage, from 6.25 up to 98.
Yeah.
Yes. Did you see any major difference among all these different dosage, different concentrations?
Okay, about the concentration, we have to perform all of the concentration to see which concentration is the best. As
No, which one? Which one is better, higher or lower, or doesn't matter?
The highest. It's the 98% concentration is the best.
98%, yes. So my question then will be relevant here. So do you think there's any possible effect on the host cells?
The possibility to the
On the host cells, epithelium cells, whatever cells, from our host.
we have to still perform more research for that. So this is that will be our next research about.
Thank you, Dr. Kano. We have one more question in the chat box from Dr. Hendrawan. So he wants to know why you select these 2 species in your biofilm model.
Okay, thank you for the question. So we choose Streptococcus mutans and then Lactobacillus acidophilus as our bacteria in this research because it's the most common bacteria we find in carriers. Yes, we do know that some of Lactobacillus species could be oral probiotic to decrease oral streptococci, but some study says that Lactobacillus acidophilus itself will work synergetic with Streptococcus mutans to perform carriers. So it will help each other to make the carriers more worse, like that.
Okay, so do you have any more questions for Dr. Kano?
One last question for me. Any reasons why you chose to have 0.2% chlorhexidine as your positive control?
Okay, thank you for the question. So we chose chlorhexidine 0.2% because it's the golden standard to prevent the carriers that we use in these days. So it's like the golden standard that has been spread in the use of the prevention.
You use chlorhexidine to prevent carriers?
Yeah, to prevent the carriers, to prevent biofilm. So it's before the carriers turn into the cavity, the chlorhexidine 0.2% is just to be an antibiofilm. As an antibiofilm.
Interesting, thank you.
Thank you.
Okay, so now we move on to our next presentation. So let's welcome Dr. Varavan.
Yes, I'm here.
Yeah, please share your screen.
All right. Yeah. Can you see my presentation?
Yes.
Okay, may I start now?
Yes, please.
All right. So hello, everyone. Let me ask you one question before we start. Have you ever heard of how good bacteria can be beneficial for oral health, more specifically to root canal pathogens? So good afternoon, ladies and gentlemen, respective judges and colleagues. On this occasion, I'm Deandra from Trisakti University, Indonesia, and I'm going to talk about our research entitled Reuterin inhibits Porphyromonas universalis virulens genes in root canal biofilm model. First, let's talk on the clinical problems. So, do you know, based on global burden of disease on 2017, that 3.5 billion people worldwide population experience caries on permanent teeth, while 20 till 50% of world population experience periodontal disease. These both diseases can eventually cause root canal infection. When caries is not treated properly during its early stages, it might develop and cause infection to the pulp chamber and causing root canal infection. And the same goes for the periodontal disease. It could infect the pulp through the bacterial contamination. As we all know, root canal infection is mediated by biofilms, and Porphyromonas gingivalis, or we call it as PJ, is one of the most isolated bacteria in teeth with root canal failure. Now on this slide, you can see how PJ works on biofilm. It mainly mediated through its fimbriae, which it is one of the virulence factors, and fimbriae is being encoded by virulence genes. Those are Fim-A from the long fimbriae and MAV1 from the short or minor fimbriae. HEC-Bay gene is also required to stabilize the interaction and has critical roles in coaggregation with other bacterial species. To perform root canal treatment, it is very important to remove biofilm completely, as the infection is biofilm mediated. An antimicrobial irrigating solution plays a very important role in this treatment. Irrigating solutions like sodium hypochlorite and chlorhexidine are widely used nowadays and have been used for more than 70 years. We sure there are many advantages, but still that some disadvantages are found. Sodium hypochlorite is a highly toxic if it is exposed to the periapical tissue, whereas chlorhexidine cannot dissolve most tissues. Therefore, looking for alternative therapy here becomes interesting. So according to World Health Organization, probiotic is defined as living microorganism, which when administered in adequate amounts can confer a health benefit on the host. So what if we use probiotic as an alternative? Some studies had found that probiotic, more specifically Lactobacillus reuteri, as one of the commercial probiotics that has been proven to be beneficial. It can produce an antimicrobial compound called reuterin, which has an antimicrobial activity. In this study, we tried to determine the effect of reuterin against Vim-A, MAV1, HEC-Bay gene expression in Porphyromonas gingivalis biofilm in an ex vivo root canal model when used as an irrigating solution. Firstly, we isolated reuterin from Indonesian strain L. reuteri and L. reuteri prudentis. Then we quantified the protein concentration using Bradford assay. Then we investigated the minimum inhibitory concentration of reuterin on PJ to examine the reuterin as effective as the conventional irrigating solution. To see the potential application of reuterin, we carried out 24 premolar teeth samples, and these samples were sectioned to cemento-enamel junction, then performed cleaning and shaping and enlarged all canals to size F2. Subsequently, teeth were inoculated with pathogenic endodontic bacteria to form ex vivo biofilm model. Then teeth samples were divided into 4 groups according to irrigant types. Group 1 were reuterin isolated from L. reuteri Indonesian strain. Group 2 were reuterin isolated from L. reuteri prudentis. Group 3 were sodium hypochlorite with 2.5% concentration as positive control. Group 4 were saline as the negative control. Then each irrigant types were divided into 2 contact times, 5 minutes and 30 minutes. Subsequently, we concluded colony counting and RNA extraction, followed by reverse transcription for cDNA synthesis, subsequent to gene expression quantification by qPCR. So here are the components of qPCR and list of primers that we use for this study. We also use PEPO gene as the housekeeping gene as well to normalize our data. Now that the research were done, MIC tests showed that reuterin at 12.5 microgram per milliliter concentration can inhibit the growth of PJ. Moreover, based on the results of colony counting, On an ex vivo biofilm model, it was seen that there was a decrease in the number of PG colonies after being irrigated by reuterin with 12.5 microgram per milliliter concentration at 5 minutes time and at 30 minutes contact time as well. This has proven that as an irrigating solution, reuterin have a great anti-biofilm ability against PG biofilms. Furthermore, several genes showed significant differences in expression compared to controls after being treated with 12.5 microgram per milliliter reuterin. That these genes were VIM-A, MAV1, and HEC-BA. So there was a significant difference between reuterin isolates and negative controls in the expression of VIM-A, MAV1, and HEC-BA gene. So now let's discuss how probiotics can hinder biofilm formation. As we know, probiotics are capable of competing with pathogenic bacteria for surface attachment by altering their surroundings. And one of the mechanisms is to produce an antagonistic compound. And one of it is bacteriocin named reuterin, which is already proven to interfere with biofilm attachment and formation, as well as the mature biofilms. So as we already seen, the results showed all genes are being downregulated. So here is a previous study by Ishikawa et al. Stated that lactobacilli overall downregulated the expression of VIM-A, which encodes the major fimbriae. So downregulated or deficiency of VIM-A gene can lower the capacity for attachment in forming biofilms. In this study, also showed that MAV1 gene being downregulated. A previous study by Xinhuan Lin stated that inhibition of production of minor fimbriae in PJ may restrain accumulation of the organism, which is required for efficient infection. The last gene of this study, HEC-B, were also being downregulated. By being downregulated, Connolly et al. Stated that rates of adherence showed a 10-fold decrease. Since HEC-B plays an important role in maintaining stable addition, even in fimbriated strains. So considering foregoing in vitro and ex vivo data as well, the results indicated that the anti-biofilm activity of reuterin against PJ may be due to attenuating the virulence genes. Hence, reuterin may develop as a potential source for future root canal treatment. Here are the reference of this study. Thank you so much for the attention.
Okay, I have a question for you. Thank you very much for your presentation. It's very nice. So I just wonder, in your conclusion, you mentioned that the further clinical study should be performed. So how you apply reuterin to the root canal?
Okay.
Yeah, how you conduct, if you want to conduct a clinical study, how you apply this antibacterial medicine or herb to the root canal? Thank you.
Okay, thank you so much, Dr. Evelyn, for the question. So the question you asked is how we actually put the irrigating solution into the root canal, right? Yeah. So after we isolated the reuterin from the alveolatory, it's actually it looks like a liquid, and then we put it with a syringe. Yeah, we're syringe that from the tip that has been inoculated with the bacteria and the biofilm has already formed, and then we use the syringe to put the reuterin irrigating solution into the ex vivo biofilm model. I hope that this kindly answers your question, Dr. Evelyn.
Okay, thank you.
Thank you, Dr. Evelyn.
Can I have I have just two questions for you, and thank you for the presentation. I'm just wondering what is the biofilm age of PG that you have used in your study? For how long you developed the biofilm?
Okay, so in my thank you so much, Dr. Ali, for the question. So for how long did the biofilms form? So we waited for like 24 hours, and so in this case, we actually trying to conduct the research for the mechanism of the reuterine. So we try to see the gene expression that contributes to the biofilm formation. And then after the biofilms are formed, and then we irrigate it with reuterine and the positive control as well and the negative control as well. And then we try to extract the RNA of the biofilm. We extract the RNA and then we do the reverse transcription to a cDNA, and then we try to see the gene expression from the qPCR method. So,
but what about the colony forming unit experiment? It was also conducted on the 24 hours old biofilms?
Yeah, to all the biofilms, right? Yeah, so we wait.
So you
like,
used like 24 hours for both experiments?
Sorry, so after we inoculated the bacteria to form the biofilms, and then the next day, after 24 hours, then we irrigated the irrigating solutions.
Okay, okay.
Yeah.
So the concentrations that you used in the QRT-PCR experiment, so what is the relation of this concentration to the MIC?
Okay, so when we want to conduct a gene expression, or because we want to see if so actually for a irrigating solution is actually we need to complete eliminating the bacteria from the root canal, right? But since in our study, we wanted to see which mechanism that reuterine tried to inhibit. So that's why we use the MIC so that we can see which gene expression, which genes that is reuterine can inhibit it. So these genes that we use in this study is actually contributes to the biofilm formation.
No. My question is that the concentration of your agent in the gene expression experiment, was it the MIC or a sub MICs?
It's the MIC, Dr. Ali. It's 12.5 gram per milliliter.
Yeah. Okay. So one final question. So you have tested 3 genes of PG. So why do you specifically select those genes to test? And do you have any information about the role of these genes in mediating the polymicrobial biofilm in endodontic infections? Like, do they contribute to the aggregation of PG with other endopathogens? Or do you have any kind of information about this?
Okay, thank you so much, Dr. Ali. So the 3 specific genes that we choose on this study, so the first gene is the FIM-A gene, is actually has a critical roles in initial adherence. So the FIM-A gene, the roles of FIM-A gene in PAGE, so PAGE can attach to a surface because the work of the FIM-A. And then the next gene we use is MAV1. It's actually for the microcolonies and maturation stage of biofilm formation. So it's like the 2 last stage of the biofilm formation. They started to create microcolonies and the maturation stage. And then for the last genes that we use is HAGB, is actually from the hemagglutinates, but these genes can eventually stabilizing the interactions that being conducted by the FIM-A genes. So yeah, doctor, so that's the 3 specific genes that we use in this study.
Okay, thank you.
Thank you so much, Dr. Ali. Hope that kindly answers your questions.
Thank you.
Do we have any more questions for Dr. Varlan? If no, let's proceed to the next presentation. Dr. Hendrawan. Yeah, please share your screen.
Okay, is my screen visible?
Yes.
So from cancers to Alzheimer's disease, the study about the link between the oral microbiome and systemic diseases gives valuable insights into the pathogenicity and the risk factors associated with it. Today, we will talk about how the bacteria in our saliva is changed in people with one of the most prevalent non-communicable diseases, diabetes. Good afternoon, delegates. My name is Devin Hendrawan, and here is my presentation. The authors declared no conflict of interest in the study, and this research was supported by 2 research grants from Universitas Indonesia. Type 2 diabetes mellitus is a global health issue affecting 426 million people in 2017. Type 2 DM affects many aspects of one's life, including the oral health status. For over a decade, dental researchers have built our current understanding about the link of type 2 diabetes and oral health status. And we now know that the type 2 diabetes mellitus increases the risk of fungal and bacterial infection in our mouth and make bad periodontal status, and also reduces the oral health related quality of life. Oral fluids can be a disease indicator in our body and the oral cavity. Saliva is one of the most common oral fluids being studied, and to be a disease assessment tool. This is a non-biological material that can be easily extracted, and it's give a potential as a non-invasive diagnostic tool in systemic diseases. Some properties of saliva that can have the potential to be used as biomarkers and bioindicators are hormones, antibodies, growth factors, enzymes, microbes, and also their products. However, the study regarding the role of the cell-free bacterium in type 2 diabetes are limited. So in this study, we aim to analyze the abundance of several oral genera in the Indonesian type 2 diabetes mellitus patient cell-free bacterium based on metagenomic analysis. And this is the first study to analyze the oral microbiome on different glycemic statuses in Indonesia. The protocol of this study was ethically approved by the Ethics Committee of the Faculty of Medicine Universitas Indonesia, and in this study we selected 63 participants with the following criteria. All participants have given informed consent prior to sample collection. The participants were divided into groups based on two grouping criteria. The first one is HbA1c levels based on the American Diabetes Association classification. The participants were classified into normoglycemic, pre-diabetic, and type 2 diabetic groups. Our second grouping criteria is based on the Green and Vermeulen Oral Hygiene Index Simplified, or OHIS, classifying the participants into good, moderate, and poor oral hygiene. The samples were then prepared by collecting the unsimulated saliva from participants by passive drilling. gDNA was then extracted, and 16S rRNA quickly to V4 amplification was done for further DNA sequencing using Illumina MiSeq system. The data were compared to the human oral microbiome database, and statistical analysis was performed. The results of our study are as follows, and 63 subjects participated in this study with these characteristics. Based on our grouping criteria, we noticed that OHIS score is generally higher in people with higher HbA1c levels. Further statistical analysis showed a strong positive correlation between OHIS and HbA1c levels. These findings are consistent with several studies that associate bad glycemic status with bad oral hygiene. Our sequencing produced 14 million sequences read, classified into 9,501 operational taxonomic units, or OTUs. The sequencing depth is fairly good, indicated by the good coverage index of 0.99 in all groups. And out of the 9,501 OTUs, we identified 2,641 in normoglycemic group, 4,971 in pre-diabetic group, and 7,353 in type 2 diabetic group. This indicates a higher diversity as with increasing HbA1c levels. Now, let's dissect into other diversity, and this is a little bit more. Here we can see an increase, a higher score in observed species count, CHA1 index, and ACE index in pre-diabetes and type 2 DM groups. These results indicate a higher richness of the oral microbiome in higher HbA1c levels. Now, moreover, the Shannon and Simpson index showed a higher evenness and diversity in prediabetes and type 2 DM group, as observed in the species abundant rank as well. And the phylogenetic diversity analysis using phylogenetic whole tree showed increasing value in prediabetes and type 2 DM groups. And the rare fraction groups also showed a steeper line in prediabetic and type 2 DM groups, implying that this is phylogenetically more diverse. The principal coordinate analysis, or PCOA, showed a dissimilarity tendency between groups confirmed by the NLCM and MRPP analysis. The samples are also able to be clustered as follows. And previous reports have shown that the oral environment is changed in diabetic patients, such as lower cell-free flow and higher cell-free amylase and glucose levels. And these changes in bacterial structure could be an additional explanation for how the oral pathogenic processes happens in diabetic patients. Now, we further analyzed the composition of the microbiome, and here you can see the relative abundance of several oral genera in those three groups. Here you can see that the top 10 genera in all groups have the same members with different compositions. However, when analyzing this data, we come across our first limit of study, which is the unavailability of bacterial load data. Therefore, the relative abundance is the only retrievable data with no absolute abundance information. So from statistical analysis, we are able to note 5 genera that are significantly and consistently lower or higher as the HbA1c level increases. Out of the 5 genera, we observe lower relative abundance in 2 genera, the genus Streptococcus and genus Haemophilus, and higher relative abundance in 3 genera, the genus Haemophilus, genus Prevotella, and genus Veillonella. The lower genus Haemophilus abundance could be correlated to the lower abundance of genus Streptococcus because one study suggests that they provide the V factor for this genus. And the most dominant change in bacterial composition is the lower abundance of genus Streptococcus. And this feature is similar to a study in Thailand, showing lower Streptococcus relative abundance in potentially diabetic group. However, this characteristic is not exclusively observed in type 2DM patients, but also in periodontitis patients with shifted relative abundance towards periodontopathic genera. A study also showed a lower Streptococcus abundance in patients with bad periodontal status and bad glycemic control. However, the most significant reduction happened in people with periodontitis and type 2DM. This pattern is also observed in this study with higher abundance of genus Porphyromonas and genus Prevotella as well. However, we come across another limit to our study, which is the unavailability of detailed periodontal records. Therefore, correlation analysis could not be performed. So to conclude all of the findings in this study, the main feature of the salivary bacterium in Indonesian type 2DM patients are lower Streptococcus and Haemophilus relative abundance, higher Porphyromonas, Prevotella, and Veillonella relative abundance, and higher richness, diversity, and phylogenetic diversity. To provide a concrete comprehension, absolute abundance study, pathogenomic study, salivary profile analysis of samples, and also analysis including the oral microbiome and virome should be conducted. We hope that this study can complement our current understanding of the changes happening in the oral health of diabetic patients and to provide a better, more precise and personalized oral care for diabetic patients. That was the end of my presentation, and here's my contact information, and please do not hesitate to contact me through email or LinkedIn. I'll be very happy to connect. Thank you very much.
Okay, thank you, Devin, for your presentation. Do you have any questions for
Okay, so may I have one question?
Yes.
So very nice presentation, yeah. So I just wonder, have you ever compared the microbiome data with the clinical parameter? Because I'm very curious about the periodontal status of your the patient with diabetes group. So have you ever make comparison? Thank you.
Thank you very much for the question. This is actually a limit of our study because this data was actually collected by a trained personnel, not a dentist. Therefore, they cannot perform periodontal examination. That's why we are actually lacking periodontal status data, but that should be another addition in our future study as well. Yeah.
Okay, thank you very much.
Thank you very much.
I just have one question. Thank you for the presentation. I just want to know how can the findings of your study be translated in clinical way to design like a new kinds of treatment for the diabetic patient? How can this be translated in such a clinical way?
Thank you very much, Dr. Ali, for the question. Now, we see that there's actually an increasing, a higher diversity in diabetic patients, but we must note that other than providing protection from other pathogenic bacteria from outside to reside in our oral environment, this diverse condition could also be a reservoir for antimicrobial resistant gene bacteria. So that should be noted. This is a possible way that the diabetic patients have it's hard for the diabetic patient to combat infections. And other than that, we can also plan for other probiotics treatment maybe in future studies. We can look for that as well.
Okay, thank you.
Thank you very much, Dr.
So any other questions? If no, let's thank Devin. And now let's welcome our last speaker, Dr. Devin from University Indonesia.
Can you guys see my screen?
Yes, we can see your screen.
Okay.
It's not in the full screen. Yeah, now it's okay.
Okay, let me start my presentation. Good afternoon, everyone. My name is Devin, and today I'm going to present to you about my research about oral Veillonella profiles associated with stunted groups in Indonesia's children dental biofilm. Let me say a few words about my research breakdown. So stunting as an impaired growth and development as a result of poor nutritional status is one of Indonesia's most concerning children health problems. And based on the National Basic Health Research 2018, the highest stunting cases were found in the province of East Nusa Tenggara in the 5 to 12 years age group with a prevalence of 41.1%. And children are stunted if their height for age is more than 2 standard deviations below the WHO child growth standards median. And in short and long term, stunting can result in a decreased intellectual development, decreased physical abilities, and increase in symptoms of depression. Based on previous research and studies about oral health and dental health, one of oral bacteria related to nutritional status is oral Veillonella. Oral Veillonella is a gram-negative bacterium that actively colonizes with Streptococcus and act as non-triggers for dental biofilm maturation and could only be found in oral cavity. Until now, there are 7 species of oral Veillonella that have been identified. Previous studies stated that there was a relationship between oral hygiene index simplified, the population of oral Veillonella in genus level as a component of early colonizations of dental biofilm, and nutritional status. It was shown that there was an increase in the population of oral Veillonella in children with poor OHS, and also there was a tendency of worsening OHS in children with poor nutritional status. But the relationship between the number of oral Veillonella both in genus and species level and stunting as a result of poor nutritional status had not been known. So therefore, in this study, we want to fill this link, and for the long-term goal, we hope that this research can be used as a pilot study to explore the potential of oral Veillonella as an early bioindicator of stunting in children. So in this study, a total of 60 children consists of 36 males and 34 females, aged 6 to 7 years old, were selected based on highest stunting prevalence group in Indonesia. First height for age measurement were performed based on WHO standard and divided the result into normal and stunting group. And then we performed OHS assessment and dental biofilm were collected. We decided to collect dental biofilm from mandibular first to molar because this tooth is just erupted and exposed to oral environment. In the laboratory, the homogenized samples were extracted. Then we measured the DNA concentration. Then we performed real-time PCR with absolute quantification analysis based on Veillonella species standard curve that we made using ATCC with regression more than 0.985 to quantified species. For a result, the demographic data can be seen on the graph. Based on real-time PCR absolute quantification, all 7 species of oral Veillonella were detected. This result is in line with previous research in Japanese children that oral Veillonella can be found in early colonization of dental biofilm. And on the right picture, we can see that overall number of oral Veillonella species are lower in stunted group compared to normal group. This result confirms that most likely there's the changes of the GI tract microbial community in stunted children. And here we can see that oral Veillonella population has smaller number in stunted group. An analysis using man-Whitney test showed that only number of oral Veillonella, which is Veillonella denticariosi, in the stunting group was less than normal group with a statistically significant difference. analysis between the number of 7 oral Veillonella species with OHIS group. In here, we can see that there was a tendency of higher oral Veillonella number in poor OHIS group, and this result is in line with previous research 2018. But Veillonella denticaryosi was found to decrease progressively with poor oral hygiene. And statistical analysis showed that only Veillonella denticaryosi and tabatuensis were statistically significant. An analysis within oral Veillonella with OHIS in normal group showed that there was a tendency of higher Veillonella number in poor OHIS, but only denticaryosi that also found to decrease progressively with poor oral hygiene. And analysis between oral Veillonella species in stunted group with poor OHIS, the oral Veillonella number in good OHIS is lower than moderate OHIS, but in poor OHIS, there was a tendency of lower Veillonella number compared to moderate OHIS. So this result shows that both in normal and stunted group, the increase of oral Veillonella was proportional to the worsening of OHIS, but in poor OHIS, the result is quite different. In normal group, poor OHIS was higher, has a higher number in oral Veillonella, but in stunted group, we can see that decrease of oral Veillonella number happening. So it shows that oral Veillonella composition differs between the two groups. So basically, the underrepresentation of oral Veillonella in stunted children with poor OHIS could be caused by several factors. The first one, the result of shows that microbiota changes happen not only in lower GI tract, but also in oral cavity, so upper part of upper GI tract. And second, we can or hypothesis say that oral Veillonella metabolism as a presumably practical importance to support the persistence of periodontal pathogens, and Veillonella are considered the bridge species due to their ability to stimulate the growth of numerous organisms through metabolic complementation, and it played a crucial role in removing toxin, organic acid, metabolic waste from the biofilm communities and create good environment for other bacteria, especially here the periodontal pathogens. So in poor OHIS, there is a high risk of periodontal inflammation and metacolonization of dental biofilm, which leads dominant colony shifting from oral Veillonella to periodontal pathogens, where both this bacteria needs the same environment to develop. And this local inflammation also supported by a tendency of GI tract inflammation caused by the systemic condition. And here is the stunting condition. This research had several advantages, including the novelty of nature of this study, which includes the first time usage of real-time PCR machine to analyze the oral Veillonella at species level, and also the first time analyzing the relationship between number of oral Veillonella, stunting status, and considering the OHIS value. But there are some limitations, including the lack of previous research on oral Veillonella in genus and species level, making it quite difficult to find references. And because stunting is a chronic syndrome, a longitudinal rather than cross-sectional approach would have a lot to assess a causal relationship better between oral Veillonella and stunting. So in conclusion, our study highlights three major findings. The first one, Veillonella denticaryosi was found statistically different in relation with high forage and OHIS group. And second, the increase of oral Veillonella are proportional to the worsening of OHIS, except Veillonella denticaryosi. And last, there was a tendency of higher oral Veillonella number in moderate OHIS, but lower in poor OHIS and stunting group. For the final notes, further studies are still needed in confirming oral Veillonella possibility to be considered as a clinical bioindicator of stunting status, and to observe the compositional changes of other bacteria, especially periodontal pathogens bacteria. Studies using 16S rRNA sequencing analysis also needed. Well, that's all my presentation. Thank you so much for your interest and attention.
So thank you for your presentation. Do you have questions for Dr. Deven?
I just have one question for Dr. Deven. You collected the biofilm from what tooth?
First permanent molar.
First primary molar.
Well, for first permanent molar.
First permanent molar. So the age group of the children were, what is the age group?
6 to 7 years old.
Okay, 6 to 7 years old. So can you explain more about the stunting status and what is like some background information about the stunting status in children?
Okay, so actually Indonesia has a lot of children with actually not only moderate stunting status, but also severe stunting status. And we see that on previous research, poor OHIS may lead to stunting and underweight status. And in children with poor OHIS, they undergo disturbances and discomfort while eating, which greatly affects their eating habit and nutrition supply. And in Indonesia, it's just a big problem that we can't solve for many years. So we try to find what can we do from the dentistry prevalence and to actually overcome this problem.
So and how do you find the findings of your study? I mean, how these findings can help to improve the oral health status in stunted groups?
Okay, so I want to say that the only reference that the only reference or previous studies that helped my research is from the Mashima Sensei from Japan, because he did oral Veillonella research for like many years, and he only identified 7 or now maybe 8 oral Veillonella species, but we haven't found any correlation between oral Veillonella and other bacteria or other systemic condition, except oral Veillonella being considered as a breed species, because without the presence of oral Veillonella, the biofilm maturation, the dental biofilm maturation won't change from early to middle or late colonizers. Maybe that's what I can explain.
Mm-hmm. Okay, thank you.
Thank you very much for the questions.
So I have one question. Did you collect information about the oral health conditions of the children? For example, is the caries being considered when you are collecting your sample? Because we know that ECC actually they can change the proportion of the microbiome in the oral cavity.
Yeah. We did collect the other data for this, but in this research we only focus on finding the. We want to focus on finding the oral Veillonella profiles, but we also have that data, so we need that for further studies about this. Thank you for your question.
Okay, so we have. Do you have any more questions?
There's one questions in the chat.
So that would be the end of this session. And I would like to thank all the presenters and participants for your contribution. And hope you enjoyed the discussion this afternoon. And please go ahead to attend the sessions in the other session halls. So I'm now closing this session. Okay, so have a good day.
Thank you.
Good day. Thank you.
Thank you very much.